Good morning, guys. Welcome. It's Monday. Hopefully that's not super disagreeable with you. If you haven't done so already, if you could let me know in the chat that you are here. Kim, you can use informal language. It's probably a good idea to try and use as much vocabulary as you can, like biological vocabulary that you know. But if you're using informal vocabulary, that's fine. As long as the definition is accurate, using your informal vocabulary, it still has to have all of the important components of what defines that particular term. But if you're using more informal language to explain that's not a big deal. Show up later. We'll get that later. Okay. Let's talk about what we're up to today. So, actually, before I get to that, does anybody have any questions from yesterday? From any of the content that we covered yesterday? I will take that as a no. If you do, you can obviously just post in the chat anytime. One thing I wanted to mention really quickly is at this point, we've actually covered most of what you need to know in order to do the... Oh, okay, I'll get you in a second there, Jaleel. We covered most of what you need to know in order to do the diversity unit vocabulary list. By the way, if you haven't yet done the student parent information survey, I went through all of the surveys that I got. I have 24 of them out of 34, I believe, which means I'm missing 10. If you could make sure that you do that today, I still need to compile information for, I think, 10 additional students. So there's a good chance that one of those students is you. So if you haven't done it yet, make sure that you're doing the student parent information survey at some point today. You don't have to do it immediately, but today, please. I'm going to take a second look at it after block 3 today, and hopefully the rest of them will come in at that point. Anyway, I wanted to mention the species spotlight assignment. Like I said, you have most of the stuff to do this already. You'll notice that there's a breakdown here of what you need to have for each section. This is basically for each slide. Where it says a visual life cycle of your species, keep in mind that you can have that extend over 2 slides if you want, if it doesn't fit on 1 slide. Note that it says that you need to show your life cycle of your organism diagrammatically. The idea being here that you draw this. You can find a blank life cycle with no textual description and then add your own textual description, or edit one that has text and then add your own. But you don't use a pre-made visual life cycle that you find on the internet. And by visual life cycle, I just mean with arrows indicating it starts off as a whatever, germinated seed and then becomes an adult plant and then whatever. The idea is that you're going to show me diagrammatically what its life cycle looks like. Show me, it's going to explain in its life cycle obviously how it reproduces and stuff like that. And just describe each part of a particular organism's life cycle. That's what you're going to be researching for that part. For the image of the species, here you can use a pre-made image. So you can just find an image of your species. But the idea is then on the right-hand side, you're going to have some lines coming over from different structures on your diagram or on your picture of your species. And then you're going to be describing some of the main physical characteristics of your organism, or morphological characteristics, and ideally explaining why that structure is particularly unique or important for your organism. Maybe if you're talking about a primate, you could talk about opposable thumbs, or you won't do primates because they're vertebrates. So you're going to pick an invertebrate, something that does not have a backbone for this project. Vertebrates is easy. Vertebrates is too easy. You're too familiar with them. You know how to make a dichotomous key at this point. So in order to do the dichotomous key for your species, again, this is your species, you can choose whatever you want, you need to pick 5 other species from the same genus as your species. Now, depending on the species you pick, there may not be 5 other species in your genus. So that means you have to go up another level of taxonomy, okay, another taxa. Which would be the family. Family is above genus. So then you look for 5 other species that are in the same family as your organism. And you know what? If there's not 5 in the same family, then you need to go up another level of taxonomy. You probably will find 5 in the same family. It would be unlikely that you wouldn't. But just in case, you just keep going up levels of taxa until there are 5 other species that you can find, okay? You're going to find pictures of those 5 species. So you got your species and 5 others. That's 6 total. And then you're going to create a dichotomous key using the exact same tools that we did in the last class for how to identify your particular species from those other species. And make sure that I can actually see the things that you're choosing from the pictures that you choose. There should be in total 6 pictures, right? One of them will be your species. And then each one should have a name under the picture so that I can... And some people do this on the previous slide, so they have their pictures and the names. And then on the next slide, where they have their dichotomous key, they only have the names at the terminus at the end. Or even on the previous slide, if you say, you know, like, this is, you give the species name in the picture, and then you label it species 1 or species A. And then that you could put at the terminus at the end of the dichotomous key on your next page. That's fine too, if you need more space. So just make sure that I can actually see the physical characteristics that you're choosing to make your dichotomous key. I mentioned that behavior is part of morphology. That is true. However, for this project, please stick to physical characteristics that I can see. Because if you say, oh, well, you know, like this particular organism has a loud mating call that is a whoop, 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 or something like that, or I don't know. That's like not something I can tell from the pictures. Ideally, I don't have to do a ton of research to be able to mark your presentation. So... if you wouldn't mind, just you stick to physical characteristics. It certainly makes it easier. It'd probably make it easier for you as well. If you are doing a bacteria, which is an option, you can choose bacteria. I don't recommend it. It's harder to do that. The physical characteristics in terms of just looking at bacteria they're going to be identical. I mean, not actually, as you'll see today, because we're going to talk about bacteria today. They're not all physically identical, but there are many, many different species of bacteria that are physically identical to each other. So you can't just purely go by physical characteristics in that case. I have seen people do bacteria or archaea, which you can, but then you need to include things in your dichotomous key like stains purple under this dye, yes or no. Or converts this metabolite into this metabolite, yes or no, or whatever. Something along those lines. You can do that. If you do that, that's going to require more research on your part. I will also have to do the research to make sure that it's correct. While I do have a little bit of micro background, I'm not, well, I don't think anybody is proficient in the biochemistry of every microorganism. So, that's fine though. That's fine. So if you want to go that route, you can. It's just more complicated. So I don't recommend bacteria or archaea, but it's up to you. It just makes it harder for you. I do mention on here that if you are going to use an image from the internet as part of this, that you should cite it using APA format. There are a couple different ways you can do that. The easiest way for you is to just use a Citation Google Chrome extension called Cite This For Me. There are actually several of them. And then when you have, like, for example, the image on screen, you can just click that button and it will create an APA citation, which is probably the easiest way. Or you could just, you can Google APA citation, and then it will give you, like, for example, here, let me actually find you an easy tool to use here. Citation machine is a good one. You can use that. So this is easy because you can pick the kind of thing that you're citing. Like let's say you're citing just a picture, sure, image. I just saw image, digital image. Okay, and then it gives you the information that you can find potentially about this image. You know, you won't always be able to find all these. For example, it says title, you know, if any. It may not have, the image may not have a title, but you include all the information that you can find, and then you click complete citation at the bottom, and it will give you an APA citation. That's the easiest way to do it. I recommend doing that or using a Google Chrome extension. Just make sure that it's set to APA. I think this is APA. Yeah, it says right at the very top on the tab there, APA. So there are various citation systems. MLA is usually used for the arts, like if you're citing a English book or like a fictional book, or you're doing research in history or that kind of stuff. They have a separate system that they use for citations called MLA, and the most common one for science is called APA. I should mention that if you are getting into grad studies, so do you have to cite our sources for the no? So for the vocabulary list, those definitions are going to come from you. You are going to read source material, whatever, the textbook, wherever you're getting your information about a particular definition, and then you are going to give your understanding of that definition in your own words. So there's really nothing to cite there. It is your own understanding of what a particular term means. You're not claiming that idea for your own. I know that you didn't invent the term. And so a citation really isn't necessary there. When you're drawing on information in a research project, the reason that you cite is that you are indicating that you have found this information at a reliable source. In other words, it's not just coming from like what you heard from your friend or whatever about a particular thing. You have to indicate where that reliable source exists and give credit to it. And that's found all across academia. You have to do that. And that's to ensure that you don't have people just making stuff up and writing it down, because that, I mean, you could easily do that. You could just you know, make your creature like the blarg and just like make up a whole bunch of stuff about it. If there was no citation necessary for any of that, there's no way to check if that's made up or and it's a fairly low consequence here in your project in grade 11, but I mean, if you're creating a research paper that you're writing in grad school or something like that, like that's part of academia. Other people will cite your paper, so like you can't make stuff up. You have to. It has to be guaranteed true, or at least true to whatever source that you're citing. So anyway, no for the vocabulary list. Should we attach a separate document for bibliography? No. For this particular assignment, your works cited or your bibliography is just the last slide. So I think I write that right in the assignment. Yeah, a works cited slide. Yeah, so it's your last slide. That's for this assignment. You are going to have a research document that you're going to use for your presentations. That's for the other units. You don't do that in this unit. And we'll talk more about that when we get there. But yeah, there is a separate document that you're going to be submitting for other assignments, but not in this particular one. And do we have to cite images? Not in the vocab list. I'm not that concerned in the vocab list because you're not claiming that information as your own in the vocab list. Plus, all the vocabulary that you're going to be putting in there is stuff that I'm familiar with. I mean, I'm teaching this course. I should be familiar with all of the vocabulary from the course. So you're not claiming it as your own. It's not something that I need to look into to see if that like is correct for a particular definition. I know what the words mean. It's more important, like I said, when you're doing a research project, or if you're writing a paper, like your own research paper, and you're talking about the already existing body of knowledge on a particular subject. In that case, you have to give credit to who established that particular piece of knowledge in the first place. But again, not in this vocabulary list, not necessary. For the species project, can we use online images for the life cycle? You can, but the image that is used can't be the complete life cycle. And by that I mean it can't be diagrammatically and textually the entire thing. If it's just diagrammatic, it's showing pictures of the life cycle and you're adding in your own numbering, like step 1, and then you have your description to the side, and step 2, you have your description to the side. That's fine. But if you're finding an image that is the complete life cycle, that has the steps, they're all explained on the image, then that's not your work. Then you didn't create the life cycle component of that. So it's fine to have an image of the life cycle, but not to have the image be the complete textual and diagrammatic life cycle of your organism. Hopefully that explained it. So it just needs to be, the majority has to be your work. The visual part can be from the internet. Can you do my project on type of caterpillar? Sure, that's an invertebrate. Pick the type of caterpillar that you like. Absolutely. Okay. Oh, yeah, that's what I wanted to point out here. So like I said, there are a number of things that you can actually already do here. For example, you can discuss the various taxonomic levels of your species. Notice that I ask you to do it in table format on a slide where you have domain, kingdom, phylum, class, order, family, genus, species. Then you explain what are the actual classifications for each of those levels. And then there should be a third column, not shown here, but there should be a third column that explains why it fits into each of those particular groupings. So if it is a eukaryote, domain eukarya, why? What makes it domain eukarya? If it is kingdom Plantae, what makes it a plant? If you're not sure about the kingdom part, we're going to be going over that stuff together over the next few days. So you can leave that part out if you want. But then phylum, class, order, family, genus, species. We're going to be talking about, or you're going to be researching why it fits into each of those particular groups. Okay, that's part of the project. Here I've got the diagram with the lines to the right giving some structural features of the particular organism. Again, you could already do that part if you wanted to. The life cycle part, you're going to do that as your own research. You could definitely do that. Here I haven't created my own textual description of this life cycle. So this is just a very basic outline of what you need to include. This isn't showing what a complete one would look like. And then the last part here, this idea of a dichotomous key. Again, you know how to make a dichotomous key at this point. So you're welcome to create that already. Like you're ready. If you are looking for taxonomic information, where was I? Here. If you're looking for example, you don't know what the kingdom, phylum, class, order, family, genus, species is for your organism, there are two services on the internet, the Animal Diversity Web and the International Taxonomic Information System. Both of these are large repositories of taxonomy information. They index organisms from all the different kingdoms. So you can search, I think at least one of these has a common name search, where you can search by common name, and you can also search by scientific name, and it will give you the taxonomic information on a particular organism. It doesn't have every organism ever that has been identified in it, but both of them are actually fairly good. They're quite extensive, and so you probably can find most of the taxonomy for your particular organism there. And it's quite good. Like it lists kingdom, phylum, class, order, family, genus, species for a particular organism. So anyway, I put those on there because they are great resources to help you find taxonomic information. I'm mentioning this in advance. You can start this project anytime you like. It's due the day after the end of the unit. So this unit would end on Wednesday, I believe, is the last day. You'll get a whole block. I think it's block 2 on Wednesday to work on it. But then it's due the next day at midnight. So it doesn't come immediately due, but some people find that a little bit of a tight turnaround. So if you want to start it before then, you're welcome to do that if you have extra time. Okay, I'm going to stop talking about this now and get to today's content. So I just wanted to make sure everybody was clear on the expectations for the assignment. So we are on day 3. Hopefully you found that at this point. You're at the live stream, so you should be okay. In block 1, there are 2 main topics that we're going to discuss. So I'm going to start by discussing prokaryotes. We're going to go through the physiology of bacteria and archaea, which are the 2 types of prokaryotes. There is, and this is what you would do afterwards, once we go away from the lesson component here. There's a brief summary video here on prokaryotes. It's actually excellent. A little bit of it is review from the note, but there's a bunch of new stuff in there as well. And it's an excellent review in general of prokaryotes. There is a really cool TED Talk that I found last quad on quorum sensing. And quorum sensing is all about how bacteria can communicate with each other, and they can decide whether there are enough bacteria for them to change their particular behavior. It's a really fascinating idea that basically bacteria can wait until they have a large group together before they will do a particular thing. For example, cause illness or produce a particular protein. And the TED Talk, I believe it's like 16 or 17 minutes long, but it really is fascinating. It's exactly the kind of thing that you'd watch if you were taking a micro course in university. It's a TED Talk, so the terminology is very accessible. It's definitely like at the right level, but it's really fascinating. So I highly recommend that. Then you complete question 4. Question 4 is in the course notes. Again, those ones don't have to go in your homework document. Anything in the course notes doesn't go in your homework document. And then you complete analysis questions on page 53, number 3 and 14. Then we're going to have a discussion about viruses. So I'm going to do both of these lessons together at the beginning, and then I'm going to let you come back and do part 1 and 2 here, the additional content. So we're going to discuss a little bit about viruses, what their structure is, how their life cycle works, just the basics really about what a virus is. Viruses are really cool. Well, I don't want to preempt myself here, but they're very different from living things. to put it simply, they aren't really alive. Anyway, we'll get to that in a sec. But there are 2 videos here. One of them is a summary of viruses that, again, reviews a little bit of stuff from the note, but expands on several of the things from the note, especially the life cycle component. So I recommend that one. And then the second one here, so cool. By the way, I love this YouTube channel. It's called Clearly Speaking. This YouTube, they have a lot of excellent videos that are very well animated. And this one talks about where do new viruses originate. COVID is they talk about COVID in this particular presentation, but it's not specific to COVID. Because, you may be wondering, COVID obviously didn't exist, or COVID-19 specifically did not exist before 2019. So where did it come from? Did it just appear? It's a really good question. Why do we suddenly get these appearances of new viruses? And obviously the answer is evolution. I don't know if it is obvious, but the answer is evolution. And this video talks about how that happens. What is the evolutionary strategy of viruses, and why do we get new strains of particular diseases popping up from time to time? Anyway, great video. Goes over a lot of the structure of viruses that we talked about, that we're going to talk about in the note, and then expands on it in a way that is totally relevant to you. I'm sure you've been thinking about COVID at least a few times. Anyway, the reference here is fantastic. So I'm just going to leave it there. But again, great video. At that point, you can finish any of the outstanding questions from the course notes. There are a couple. I mentioned them as we go through the course notes in the virus section. They're quick little research questions. Last quad, because we're going to talk a little bit about vaccines in the virus section. Somebody asked me about whether vaccines cause autism. Interesting question. That is an idea that has been in the public consciousness for a long time. And it is due to one of the most serious errors that has ever been published, and likely from faked data, which once you publish something like that and that becomes part of the public consciousness, even though that data has been disproven and that particular article has been rescinded, unpublished, removed from publication, the damage is done. It's like once something enters the public consciousness, it doesn't matter if you disprove it subsequently. It does in the scientific community, but for some reason in the public consciousness, it's very difficult to unsay something if you find out that the The evidence does not actually support that. Anyway, the article here talks a little bit about what happened in that particular instance and why that idea exists in the public consciousness, even though, by the way, it is not true. There is no evidence whatsoever that that is true. But anyway, if you're interested in that at all, there's an article there that talks about the controversy surrounding it and where that idea came from. Then, in Learning Block 2, by the way, this sounds like a lot. I realize that it is. The next couple days are quite busy. I've had to pack the diversity unit into quite a small area. So anyway, during Learning Block 2, we're going to go over together the different types of protists that exist. So we're jumping to a completely new kingdom in Learning Block 2. We're going to talk about protists. Protists are so crazy. They are the wacky uncle of the kingdoms that just has all these zany things in it that are really strange. So we're going to talk a little bit about that diversity in the lesson. This video talks about specific examples of species that I just can't talk about all of this stuff during the lesson component. So this video is great. It has a bunch of pictures and talks about some of the strange diversity that exists inside of protists. So again, great video to watch as a follow-up. You're going to complete the summary table of 3 protists on page 13. If we were in class, unfortunately, we would normally, it's not unfortunate, but it's unfortunate now that we can't look at these under the microscope. So this is normally the time where we would do a microscopy lab and we would look at these 3 protists under the microscope, observe their behavior and their anatomy. Unavailable. I actually, I brought home a digital microscope from school so that I could hook it up to the stream here and show you some microscope images of things. Unfortunately, I'm not able to order the protists and have them sent to my home. They won't send them to my house. I don't know why. They're a school supplier of protists. I wonder if I could get some pond water from somewhere. I might be able to do that. If I could, maybe tomorrow I can find some pond water and we can take a look at the organisms inside. But it won't be these specific ones. The idea was that we were going to look at 3 specific organisms. Anyway, you're going to research them on your own. You can find pictures of them and video on YouTube. It's not quite the same as looking at them yourself under the microscope. But anyway, we'll do the next best thing. And then there are some analysis questions that go along with that. As usual, during Learning Block 3, or you can do it as you go, you add wording to your vocabulary list, and if you have any extra time, you can work on your assignment. But you might not have extra time today. Today's quite a busy one, and I will be in the Google Meet during Learning Block 3 the entire time. Thanks to everybody who came to visit me, by the way, last class in the Google Meet. It was nice to see you, and you're welcome to drop by anytime, even if you just want to share some interesting thing that you found out about biology or really any science. I'm interested in everything. So, okay, let's get to the note here. I'm not gonna say I'm gonna rush through it, but I'm gonna go through it at a fairly good pace. Knowing that you guys can go back and look at stuff at any time, because I want to make sure that you have time to do the parts that you have to do as well. So I'll try and keep things flowing. Let me know again as we go if you've got any questions. Feel free to type them in the chat. I don't think I've missed anything yet. So, okay, prokaryotes. We've already mentioned, and you don't have to write this down again, but they don't have membrane-bound organelles. So they've got no nucleus. They have DNA, but their DNA is just floating around inside of their cytoplasm. They don't have a Golgi, they don't have mitochondria. So when you look at these under the microscope, one of the things that you'll notice is that, first of all, the prokaryotes are very small. In order to support a large cell, like the cells that we have, by the way, like our cells, you have to have fairly complicated cellular machinery inside to support that much volume of stuff. Complex processes are required to support the metabolism of such a large structure. And prokaryotes just don't have that. They don't have the machinery on board. So their maximum size is quite a bit smaller. I wonder if I can find a size comparison real quick just to show you. Yeah, I mean this is pretty good. This is pretty good. So this cell on the far right here is 10 micrometers is pretty close to an average cell size for a human. You can't really say average cell size for a human because we have some monster cells and some really tiny ones. The red blood cell right there is the smallest cell that you would find in humans. It doesn't have a nucleus. It's quite small. So 10 microns, maybe 30 microns, maybe 3 times that size. They were getting in the range of a normal eukaryotic cell size. If we look to the left here at the bacteria in comparison, that's approximately an average size for a bacillus. A bacillus is a certain shape of bacteria. Cocci are spherical and they're a little bit smaller. But that is approximately the size comparison there. I would say to an average cell, it's 1/50th of the size, or I mean, if it's quite a large human cell, maybe 1/100th of the size. So they're quite small. And then we're also going to discuss viruses today. It's important to note that viruses are extremely small. So the average virus, and that is a coronavirus. There are different types of viruses that are different sizes, but an average virus is maybe 1/10 of the size of a bacteria. There are viruses that attack bacteria, by the way, that are considerably smaller than that. They're called phages, bacteriophages rather, and they're usually quite a bit smaller because they infect. They go inside of bacteria, so they're quite small. But a coronavirus, which is something that's typical that would infect humans, there are a number of different coronaviruses, by the way, that cause a cold or things like that. You've almost certainly had a coronavirus infection sometime during your life. They're maybe 1/1000th the size of a human cell. They're quite small. So I just want to give people a general idea for sizing here. So when we're talking about prokaryotes, we're talking about quite small cells. There are 2 main classifications of prokaryotes, as we mentioned. These are the 2 domains, which are eubacteria and archaea. Eubacteria, domain eubacteria, has 1 kingdom in it. It's weird to think of it that way. But domain eubacteria only has bacteria kingdom in it. So it's a domain that only has 1 kingdom. And domain archaea also only has 1 kingdom in it, and that is kingdom archaea. So little bit of a weird way to organize that, in my opinion, taxonomically, but they consider archaea and eubacteria to be so different from eukaryotes that they gave them their own domains. So anyway, you guys already know this. They are single-celled, so they have no membrane organelles. They are the smallest organisms on Earth. And maybe you might be saying, hey, wait a second, Wiegelmann. Didn't you just say that a virus is 1/10 or 1/100th the size of a bacteria? Yes, I did say that. Except viruses are not considered to be organisms. They're not considered to be alive. So there's a slight caveat there. And you'll see why once we talk about viruses, but that's the reason why I can make that statement. I read somewhere that the smallest human cell is a sperm cell and the largest is an ovum. Yes, so an egg cell, human egg, an ootid, they're called, is by far the largest human cell. A human egg cell is so large that you can actually see it with the naked eye. So if you like placed it on a piece of paper, you would be able to see the egg cell. I mean, that's that's tremendous. That is like absolutely gargantuan in terms of cell size. And we're in the genetics unit, we'll talk a little bit about why they have to be so large. We do actually come back to this concept. Sperm are small. I don't know if they're smaller than a red blood cell, though. That one I'm gonna have to investigate, Kim. They may be similar in size to a red blood cell. I have to double check that. I thought that sperm were larger than a red blood cell, but I may have to look into that. They're close. They're on a similar scale to each other. So, I think definitely if you include the tail, they've got to be longer than a red blood cell. They've got to be. Maybe in terms of volume, they're smaller. They don't have a lot of volume inside. That one I'm going to have to look up. I'm not 100% sure on that. But that's, I mean, you're certainly right about the egg cell. What are viruses then? You mean, what are they classified as, Trenton? Great question. If you're a virologist, you would say, I think they are alive. Well, maybe you would. I don't know. But you probably would consider them an organism as a virologist. They're in like a weird biological gray area. We would call them biologically active molecules. I don't know. I mean, like that's probably a good description of them. I'm not actually sure. They are technically classified as non-living. Yeah, biologically active molecule is probably a good way to think of it. It's the same thing as prions. Prions are in a similar classification where they're molecules that affect biological systems and are complex, but they are not alive themselves. I'm not 100% sure. I don't think that there is a classification word for them. So, anyway. Now that is also a great question. Sorry, we're getting a touch off topic, but it fits perfectly in with this course, that question, Trenton, which is, are they the oldest? We have no idea, because viruses do not leave a fossil record at all. They're too small. So we have absolutely no idea when viruses first developed on Earth. We do not have any clue when they first showed up. There is some idea in the literature and just in biology at large that viruses may have been contributing factors in causing evolution to occur. So because they cause mutations in organisms. And so they may have been an important factor in causing evolution. But when they showed up in terms of like when they first became part of the biology, no idea. We actually don't know the answer to that question at all. When you say similar to the earliest life, like I get what you're saying. Like they are simple organisms that are essentially just biomolecules that exist. The problem is they can't exist just on their own, because they have no replicative strategy, no way to reproduce themselves on their own at all. So the key to the first life on Earth is that it had some type of reproductive strategy, some way to make copies of itself in that primordial ooze that they existed in the early Earth. So in that way, they are different from the earliest organisms on Earth. But they are a collection of molecules similar to molecules that could have formed in the early Earth. RNA, DNA, depending on the type of virus. Some are RNA and some are DNA. A capsid, which is just like a protein coat. Those types of molecules can form in that environment of the early Earth. So yes, in some ways they are similar organisms, but they don't have that property of a living thing that the earliest cells would have had, or the earliest organisms, even pre-cell, in that they have some type of replicative strategy. Do viruses eat or consume nutrition of some sort? No. They have no metabolism whatsoever. I'm going to just hold off on the virus questions for 1 second, because we're going to spend a whole lesson here going through viruses. If you have these same questions when we're done that part, come back, okay? Definitely ask them. But I'm pretty sure I addressed that in the virus lesson, coming up very shortly. Okay, so anyway, despite their small size, they are the dominant form of life. Hard to imagine. But these guys make up by far the greatest biomass on Earth. They make up the greatest population on Earth because they live everywhere and on everything. So they live inside organisms. They live inside of the Earth. So even if you go down inside the lithosphere, the places where life can exist in the Earth's crust, basically there's bacteria everywhere, or archaea. They are living inside organisms. They're living on the surface of organisms. They are suspended in the air. They are everywhere in the water column of every water column, even boiling water. like you might see in a hot spring. There's going to be archaea that are living in a hot spring. So they are everywhere. You have probably 100 trillion bacteria living on and in you. You are literally covered inside and out with bacteria. Your upper GI tract, so that would be from, or at least not the entire upper GI, because you have lots of bacteria in your esophagus and stuff like that. But after your stomach acid, which kills most of the bacteria that's entering your digestive tract, from there until the end of the small intestine, it's fairly sterile. It's not completely sterile. There is some bacteria there. But after that, in your large intestine, there is a whole whack of bacteria and archaea living in there. So the inside of your gut is chock full of bacteria. Interesting trend. Maybe you could forward that to me in an email. I'd love to take a look at it. Anyway, they're everywhere. So, and because of that, I would like to say that they are the dominant form of life on Earth. They are certainly the earliest form of life on Earth. They replicate extremely fast, and they mutate and evolve extremely fast. Very likely, and this might seem counterintuitive, but we've probably only identified about 1% of all of the prokaryotic species. And the where we get that estimate from is that we keep checking. And we look at the DNA of the prokaryotes in a particular sample, soil samples, air samples, you name it. Whenever we do that, most of the DNA that we get back is new. And so we can make a rough estimate based on how much new stuff we find every time we look. We probably only have indexed about 1% of the DNA and identified about 1% of the prokaryotes that are out there. So there are so many prokaryotes, different species in existence, and we really know very little about them. We like to pretend, especially if you talk to a microbiologist, they really do like to pretend that we got this covered. Like we know what all the stuff is that grows on of this petri dish and whatever. That's not really true. That's not really true. We know about the dominant ones, the most common ones. If you leave a tray of agar, which is basically like a sugar that bacteria like to grow on. If you leave that out and you see a bunch of stuff growing on it, yeah, sure. The main stuff that grows on it every time, we can identify what that is. We know those species. If we take a scraping off of human skin or we take a sample from the gut, yeah, sure, we can identify most of the stuff that grows there. But if you just take a random soil sample, there is so much new stuff in there every time you look. We just don't know nearly as much as you'd think. It's quite surprising, actually. So, really important to remember that as humans, historically, we really, really care a lot about pathogens. So pathogens are bacteria that cause illness. Okay, or an illness-causing agent, I should say. Wow, we really obsess about pathogens. I mean, obviously, we talk about them constantly. They are the things that are the most important to humans. And because we talk about pathogens so much, we have a really weird bias when it comes to discussing microorganisms, in that we tend to discuss them like they mostly cause illness, like they are mostly pathogens. But that could not be further from the truth. There is an extremely small subset of microorganisms that cause illness. The vast, vast majority of them do not cause illness in humans. And that's really important to remember, because when you go and you buy a antibacterial soap or an antibacterial mouthwash or antibacterial this or antibacterial, because everybody's obsessed, antibacterial, I got to get antibacterial. Everything's got to kill all the bacteria. Most of those bacteria that you're killing are doing important tasks. They are maintaining the pH of your skin, helping you remove waste products, old dead skin cells, etc. from your skin. Your gut, the bacteria that live in your gut are producing essential nutrients for you, and you are absorbing those nutrients. You are literally living in harmony with your gut bacteria. So this idea that you have to constantly be killing bacteria, kill the bacteria all the time, is a crazy idea that we have gotten because we just seem to have become obsessed with this tiny, tiny subset of bacteria. I think most microbiologists, especially in university, they always talk about this, about how people are obsessed with pathogens. But this is a real poor understanding of microorganisms. So if there's one thing that you take out of this, Honestly, that is one of the most important things to take out of this diversity unit, which is that microorganisms are mostly your friends. They are things that you need to survive. They are absolutely important ecologically and for your health. So don't get that idea in your head that all bacteria is bad. That is a ridiculous concept. The problem is that pathogens, the ones that do cause illness, are bad. They really often can be very bad. And so we do have to do some things in order to remove the pathogenic bacteria. For example, if you wash your hands carefully before you eat, which by the way, you 100% should do, and that's to remove pathogens. And should you do that? Yes, you absolutely should. Because yes, pathogens are still real, and they absolutely can cause very serious physical harm to you if you're not careful. So I'm not saying that you need to ignore pathogens, but you just shouldn't think of microorganisms as being all pathogens, because that's ridiculous. It's a very small percentage that are pathogenic. Okay, what would happen if the human body was completely rid of bacteria? Great question. I think it would cause extremely poor health. There has been some research that goes into what happens when people chronically use antibiotics. So a lot of people have to take them, long courses for various illnesses. If you have a highly depressed immune system, you have you're experiencing full-blown AIDS, or anyway, there's many illnesses that depress the immune system. You're having chemotherapy or whatever. In that case, it's very difficult for your body to actually live at harmony with the microorganisms that live on it. In that case, you have a very low ability to regulate the conduct of those microorganisms. And many of them that would never be pathogens under normal circumstances can become pathogenic to you. In other words, you get overgrowth of bacteria that normally you would easily maintain on the surface of your body. So there are cases in which you would need to take antibiotics for an extended period. You have a depressed immune system or whatever. However, it causes digestive problems, nutritional deficiencies, skin irritation. There's all kinds of problems that come from maintaining a sort of bacteria-free environment on your body. They are important in terms of maintaining your health. That being said, again, you do want to limit the amount of potential pathogens that you're ingesting. So washing your hands and things like that are, of course, important. But anyway, no. Oh, sorry. That wasn't what I meant to imply, Trenton. What I meant to imply was that there are people that have to maintain a very low population of bacteria on their bodies because they have something like AIDS. And that maintaining that low population of bacteria has its own consequences, its own side effects, not the same side effects as AIDS. The consequence of having something like AIDS is that you have bacterial populations that grow out of control because your body can't maintain proper populations. What happens when you have no bacteria is that you get things like skin irritation, digestive problems, nutrient deficiencies, because all of the jobs that those bacteria were doing, they're no longer being done. However, if you have to have that, it's preferable to have those side effects than to die from an overpopulation of a bacteria that would normally be healthy for your skin. So it's not like there's a choice. You do have to do some type of immune system management of your body, and if your body can't do it, you do have to take antibiotics, maybe in some cases. Anyway, so sorry if I confused those two. I wasn't trying to do that. Okay, I'm going to keep going here. Lots of pathogens out there, though. Hopefully you got these down. Bubonic plague, bacterial meningitis, E. coli, salmonella, leprosy, tuberculosis. Those are all caused by populations of eukaryotes. All of those, by the way, are bacteria and not archaea. Off the top of my head, I cannot think of a species of archaea that is pathogenic. I don't believe there are any. I will have to double check that. But to my knowledge, it's only bacteria that are pathogenic in humans and not archaea. You do have some archaea that live in your gut. Decomposers, okay? From an ecological role, they break down nutrients in the natural environment. They recycle them as part of all of the natural cycles, the nitrogen cycle, the carbon cycle. All of these things have bacteria really at their core that are breaking down all of the waste products from things and turning them back into usable nutrients for other organisms. You probably remember that from grade 9. There's bacteria in all of the natural cycles, except maybe the water cycle, but carbon cycle, nitrogen cycle, there's bacteria playing important roles there. They are important producers in that they form the bottom of the food web in many aquatic ecosystems, not so much in terrestrial ecosystems, but the majority of the oxygen in the atmosphere was produced historically and is continuously being replenished by photosynthetic bacteria that live in the ocean. And so they're extremely important producers, producing carbon compounds, sugars, for aquatic organisms. So as producers, they're extremely important. You would have talked about this as well in grade 9, but the idea of the nitrogen cycle, you have those nitrogen-fixing bacteria and nitrifying bacteria that produce ammonia, nitrate, and nitrite. Those are essential nutrients for plants to grow, and we need those bacteria to do that. That is the main way that nitrogen compounds get into soil, is through bacteria. So if you remove them, obviously there's no way for plants to get their nitrogen anymore, and they die. So in terms of soil bacteria, nitrogen fixers are probably one of the most important bacteria on Earth. They allow the nitrogen cycle to exist, and they allow plants to be alive. So we cannot have plants without nitrogen-fixing bacteria. You have some in your digestive tract right now. I've already mentioned this. This is a mixture, so some of them are E. coli, and there are other strains as well that live in your gut of bacteria. There are also strains of methanogens, which are archaea, that live in your gut, and they produce vitamin K, vitamin B12. These are essential vitamins, by the way. They also produce a number of complex carbohydrates that are used to build signaling molecules in your cells, and to build important metabolites that are used for intracellular communication within cells. They're important components of a whole host of regulatory proteins. So the bacteria in your gut serve a very important function in terms of producing nutrients, which, again, may be surprising for some people. One of the causes of gastroenteritis, having inflammation in your large intestine, is having a insufficient population of bacteria. It's often an illness that you get from taking antibiotics. It can cause inflammation in your large intestine. And what happens when you remove a large percentage of the bacteria in your large intestine is that when it repopulates, and it will repopulate at some point, you are going to get some bacteria regrowing. You may get only one particular strain that regrows, and in which case it may be something pathogenic. Whoops. So basically, you clear cut the forest, and then you reintroduced a weed, and then only the weed regrew is what happens. And so treating that illness with antibiotics is actually counterproductive. It may make it worse, because it may cause another repopulation of the same pathogen. Anyway, there's problems with using antibiotics. That's one of the potential side effects. And we use them for commercial use. So many cool commercial uses for bacteria. We make yogurt, we make cheese. If you're not into dairy products, we use it to make synthetic insulin. You can actually take the gene, the human gene, that is the piece of DNA that codes for insulin, which is a protein that your pancreas makes. Whenever you eat sugar, your pancreas releases that molecule, that signaling molecule. It's a hormone. And that causes your cells to take up the sugar out of your bloodstream, and it keeps your blood sugar from going too high. And it's essential. You have to do that. If you don't do it, you have an illness called diabetes. And if you are not able to produce your own insulin, you usually need to take exogenous insulin. Exogenous just means produced outside of the body. So we used to get insulin for diabetics from dogs, which is a terrible process. Even in the 1950s, 1960s, we did this, where we essentially raised dogs tied off their pancreas, which eventually kills the dog, by the way. And then their pancreas would fill up with insulin, and then we would remove the insulin from the dog pancreas and sell it. That's how we got insulin, because dog insulin, it's mammalian insulin. It's very similar molecularly to human insulin. And then people could inject that insulin if they were diabetic. Before that, if you were diabetic, you just died. So, I mean, before that, there was no way to treat people that were diabetic before we were able to get it from dogs. There just simply was. You would just go into a coma and die. So, it's a step up. but now we can take that same gene and we can just give it to some bacteria. I think we use E. coli for that purpose. And then the E. coli, we can just grow them in a vat and they will make insulin for us. They're like little factories that produce insulin. And we can do that with all kinds of stuff, by the way. We can use it to make plastics, and there's all kinds of things that you can get bacteria to make for you if you give them the right genes. So anyway, there are tons of commercial uses for bacteria. Maybe you hear about this in the news sometime, or about there's like an oil spill, and somebody has come up with a strain of bacteria that can break down crude oil and produce some non-dangerous byproducts. And then that could potentially be used in an oil spill or whatever. I mean, you hear about these things all the time. There are lots of potential uses for that technology. Cleaning up any kind of chemical spill, et cetera, et cetera. There's lots of ways to use bacteria for that. Let's talk a little bit. Okay, so we talked about some of the uses here. Keep in mind, again, the pathogens are a very small part. Lots of other cool stuff. Don't hate on poor microorganisms. Let's talk about the physical characteristics of a bacteria. So here's a typical bacterial cell. They're not all identical. There are different shapes and different surface proteins and things like that. But there are some general structures that you see on most prokaryotes. So they have a capsule on the outside. I'm going to go through these one at a time so you don't feel like you need to quickly write down everything all at once. But they have a capsule on the outside, and that outer capsule prevents water loss. It's made of a bunch of proteins and sugars on the outside. It's mostly protein layer. It, in general, will protect or help protect from viruses that attack bacteria. Those are called bacteriophages, although obviously it's not perfect. Bacteriophages can get through that capsule layer, but it's like a protective layer. It also has some protective effects from temperature changes. So you're probably already aware of this, but when liquid, like you find inside of a cell, gets really cold, it has a tendency to freeze. And when water freezes, it expands and can damage cells. Usually if it's like a human cell, for example, and the cells freeze, they lyse, they pop, and the cells die. That's how you get frostbite. Well, I won't show you pictures. It's kind of gross. But for bacteria, they're fairly resistant to that, which is why when you freeze stuff, most of the time you don't kill the bacteria that live in it. You prevent them from replicating. So when you put stuff in the freezer, they last for a long time. Food lasts for a long time because the bacteria generally can't replicate very well at those temperatures. But it usually doesn't kill them. It also protects them from hot environments, but only up to a point. Once you break somewhere around 50 degrees Celsius, most bacteria start coming apart at the seams at that point. It takes a while. If you get most bacteria up to 100 degrees Celsius, as you probably know, that's boiling water, usually in under a minute, you've killed every bacterial cell within a particular sample. So obviously really high temperatures, they're not invincible, but the capsule does a good job of providing some protection. Then there's an outer membrane layer. So the outer membrane is kind of similar to the cell membrane in animal cells in that it controls the entry and exit of nutrients and waste products. So you want to pump some sugar or some kind of nutrition molecule in. You do that, you pump it in through the outer membrane, or you want to pump some waste products out, you pump it out through the membrane. It has like these little cellular pumps that are embedded in the membrane. Right below that, there is a layer of sugar protein called Peptidoglycan. And it's a large chain of molecules that gives the bacteria its shape. So it provides it with some strength and rigidity, and it goes around the outside of the bacteria and gives it its shape. That's why you can get bacteria that are not spheres. If there's no structural integrity to the wrapping on a particular cell, it just ends up being a sphere, right? Like the surface tension will just pull it into a sphere. But a lot of bacteria are not perfect spheres, and so there's something that is keeping it structurally stable. And in most cases, that's the peptidoglycan layer. On the outside of the bacteria, there is often pili. So pili, they look kind of like hairs. They're not hairs. They're kind of like syringe tubes, needles, because they allow bacteria to exchange genetic material, which is really cool. Often they do it within the same species. So if there's a species of bacteria, there's a whole grouping of them, they can give some pieces of their DNA to their neighbors. So if one of the bacteria comes across a gene for a protein that makes them resistant to antibiotics, they can give that gene to their neighbors. The process takes time, so it's not like they instantly all become resistant. And to some extent, I believe that the process is random. So it's not like an effective way of transferring from one bacteria to many, but it does allow some type of horizontal gene transfer. Horizontal gene transfer just means sending your genes to somebody in the same generation as you. It would be like if you had a particular genetic trait and you gave that genetic trait to like your brother or like your friend, because you horizontally transferred it to them. Only prokaryotes can do this. So once you're a eukaryote, you're a complex organism, you can't just give genes to somebody who is in the same generation as you. You can give genes to your offspring, to the cells that you produce, but you can't give them to your neighbors. So that's something unique to prokaryotes. And it increases genetic diversity among the prokaryotes because they can just give their genes to their neighbors and spread genetic information around. And as we mentioned earlier, increased genetic diversity means better survival. So this is a survival strategy for bacteria. Oh, it's also true, I just wrote this as sort of a side note here, that bacteria can just pick up DNA from the area around them. They can just absorb some DNA that is just in their environment and incorporate it into their own DNA. And that process is called bacterial transformation. It's extremely useful for humans because, going back to my example earlier, if you want a bacterial cell to make insulin, you can put that insulin gene just in a pool with the bacteria, and then you heat it up, and there's some techniques to get it to take up the DNA around it. But you employ those techniques, and then it will just suck up the DNA from its surroundings and incorporate it. So transforming bacteria is something that you can do to make them build something that you want them to build. Okay, you guys have heard of a flagellum before. It is the exact same structure that you find on a sperm cell, although the exact biochemistry of how it works in bacteria is different than how it works in a eukaryote. They are similar. They're similar structures. Not every bacteria strain has a flagellum. Some of them do, and some of them do not. Some of them have no propelling structure of any kind. So remember, we're talking about like an average cell here. So some of them do, and some of them don't have that. Okay, I believe I covered it. Those are the basics. And you know what? Yeah, there's not a lot. There's no... Oh, hold on. Actually, I forgot to talk about the DNA part. Yeah, there it is at the bottom here. So the DNA in a bacteria, there is usually a main area of the bacteria. There's no nucleus, but there's an area of the bacteria that contains the DNA, and it's usually in the form of a loop, a big loop. Whereas our DNA is in strips, right, in chromosomes. You guys already know that. In prokaryotes, it's usually found as a big loop that's all kind of bunched up. And then there are often smaller loops of DNA that are also in the bacteria. Those are called plasmids. The difference between bacterial DNA and eukaryotic DNA is that bacterial DNA has no introns. Introns are sections of DNA that don't do anything, or at least they don't build anything. So in our DNA, there's lots of sections that are not coding for particular proteins. They just, so some of them regulate cell structure, or not cell structure, regulate expression of DNA. Like they tell the DNA to turn certain sections on and turn certain sections off, replicate this, don't replicate this, do this more, do this less. That introns do that in eukaryotes, but bacteria don't have that ability. So basically bacteria, their DNA is just saying, build all the things, build them all the time, and just do this all the time. There's very little regulatory DNA in bacteria. So you just don't find introns, large sections that don't code for proteins. So basically they're just way simpler. It's just a much simpler version of a cell than a eukaryote. All right, so that's the basics. Let's talk a little bit about the shapes of bacteria that you can find. So often bacteria, yes, they have a binomial nomenclature, a two-part name, but often they are identified by their shape. Okay, and you will have heard these terms used probably maybe by a doctor, but just used in common parlance here. So the three shapes of bacteria, these are the three standard shapes, are called cocci. Cocci are spheres. Bacilli, which are cylinders, I guess, tubes, little tubes. I shouldn't call them tubes because they're not empty in the middle, but they cylinders is a better way to say it. So we've got cocci, bacilli, which are cylinders, and then the spirilla. So spirilla are these spiral-shaped bacteria. Kind of cool. And so oftentimes bacteria will be named based on their shape, but keep in mind that these shapes are shared by many, many different strains of bacteria, many different species of bacteria. So we've got cocci, bacilli, and spirilla, and we also use the number of them that typically group together as part of the name, often. Not always, but often we do. So for example, a diplo, D-I-P-L-O, means two. And so you can have two of any of these. You can have two bacilli, that would be a diplobacilli, or you can have a two cocci bacteria and it would be called a diplococci or a diplococcus. Or you can have a diplospirilla. Diplospirilla is not as common. You can have what's known as a clump. So a clump is a staphylo, spelled S-T-A-P-H-Y-L-O. Staphylo just means clump. So you can have a staphylococci. Staphylococci would be a grouping of cocci. Or you can have a staphylobacillus, or staphylobacilli. And that would be a clump of these. Or you can have a staphylospirilla. An example of a staphylococcus that you may have heard of is Staphylococcus aureus. Staphylococcus aureus, or S. aureus, is a common skin infection bacteria. People get this at the gym all the time. So if you don't clean the gym equipment properly when you're using it, it's a highly contagious skin bacterial infection that people get at the gym. It's called Staph aureus. Wrestlers and things like that get this too because it can easily be transferred between people when you have skin-on-skin contact. So in sports, this is a very common illness that gets spread around among athletes. A strepto, believe it or not, the word strepto, S-T-R-E-P-T-O, strepto, just means a strip. That's pretty straightforward. And so that would be a strip of cocci, streptococci, or streptobacillus, if it's a bunch of bacilli that are stuck together. Or you can technically get streptospirilla. That exists. So an example of a streptococcus bacteria, that would be a strip of cocci, is Streptococcus pharyngitis. Streptococcus pharyngitis, or S. pharyngitis, is the most common bacteria that causes strep throat. So strep throat is not always caused by Streptococcus pharyngitis. There are other strep species that cause strep throat, but the most common is Streptococcus pharyngitis. You may have had strep throat, a very painful bacterial throat infection. I believe you can get viral strep throat as well. There's no way to treat viral strep throat. Antibiotics don't work on viruses. But if you have bacterial strep throat, then usually, assuming you don't have a antibiotic resistant strain, you can get treatment, antibiotic treatment rather. Okay, so those are those basic shapes that are often used for naming of bacteria. You'll notice, by the way, that I've only been talking about bacteria here and not archaea. In general, this does not apply to archaea. So archaea have a different naming strategy. It's usually not related to shape. So this is specific to bacteria that I'm discussing right here. There are also ways to identify bacteria based on what they eat. So again, it's not only based on body shape. Sometimes you can identify bacteria based on, or they're classified based on what they eat. If it is a bacteria that makes its own food, that is, it is an autotroph. Autotroph just means self-feeding. I got it written right there, self-feeding, autotrophic. They're usually photosynthetic, so they have a molecule that converts sunlight into metabolites for them to consume. Although there are other ways to be autotrophic. You can be a chemoautotroph, which means that you use some type of chemical means to convert that chemical into a sugar product for you to eat. There are organisms at the bottom of the ocean that are chemoautotrophs, that they use these weird heavy metal compounds that come out of the thermal vents at the bottom of the ocean to produce. Those are also autotrophic, but they're not very common, obviously, on the surface. So autotrophs usually using photosynthesis. They use light or chemicals to build their energy molecules. I just wrote the formula for photosynthesis again down there. You're probably familiar with it at this point. And then you can also have heterotrophic bacteria. And heterotrophic bacteria are bacteria that have to eat things. These are fairly common. Most of the bacteria that, for example, are living on your body or in your gut, those are all heterotrophic bacteria. They're either eating dead skin cells or parts of dead skin cells on the surface of your skin, or they're eating other bacteria. Some of them do that. Or they're eating metabolites that they're finding in your gut or whatever. You can classify bacteria based on what substrate they need in order to access their energy. So, for example, the substrate that we need to access our energy is oxygen. We require oxygen to liberate the energy from sugar molecules. That's what our metabolism does. You're probably well aware that you require oxygen to be alive. There are bacteria that also do that. The word obligate means that you need it. So an obligate aerobe means that you need air, oxygen, in order to survive. You need O2 to survive. There are lots of species of bacteria that are obligate aerobes. You would be considered an obligate aerobe. You require oxygen in order to survive. There are bacteria that require oxygen, so those are called obligate aerobes. If you prefer oxygen, that is, you work the best if you have oxygen, but you don't need it. If you don't get it, you can still survive, but you really prefer it. That's called a facultative aerobe. A facultative just means you prefer it. And I realized I actually made a mistake the other day. I called the dichotomous key, the written dichotomous key, a facultative dichotomous key. That is the wrong term. It's not called a facultative dichotomous key. It is called a descriptive dichotomous key. I don't know where I got the word facultative from. Maybe I was thinking ahead to this lesson. I don't know. But facultative means that you prefer it. So facultative aerobe means that you would prefer oxygen to survive, but you don't need it. You are not a facultative aerobe. You need oxygen. If you don't get it, you will die. But some bacteria just prefer it. Or you could obviously be the exact opposite of that, and you could need no oxygen. That is, you need an oxygen-free environment to survive. And if you get oxygen, you die. So that exists in bacteria too. If they're exposed to oxygen, they could die. That's called an obligate anaerobe. You require no oxygen. You can also, by the way, be a facultative anaerobe, although those are not as common. And that means you prefer no oxygen, but you can survive if there is oxygen. So all those possibilities exist. So we can classify bacteria like that. When you're working on your own here, I'm just going to get you to do a quick little bit of research here to list a few bacterial diseases. This is on page 51 in your text. And the species that cause them. I'm not going to go into these because I think that you may even be able to list a couple bacteria and the illnesses that they cause just off the top of your head. Again, because we obsess about pathogens all the time. I don't talk about pathogens too much here, mostly because you're the most familiar with them. But it is important to acknowledge that they exist and to know maybe what they are. So I'm going to get you to write maybe 2 or 3 examples of pathogens. 3 would be a good idea. There are some in the text. You can go back and do this later. So this is part of what you're going to work on when you're working on your own. How am I doing for time here? Whew. Okay. Oh, this is such a busy day. You guys had some good questions, though. Okay. I want to talk just very briefly here about the overuse of antibiotics. Oh boy, guys. We do this a lot. We do it less now. Oh, question. Would we put that question into our homework? No, you don't. So anything that goes in the course notes just goes into the course notes. You add this into your course notes, but it does not go on the homework document. Hopefully that answered your question, Caitlin. Nothing that is you're asked to do in the notes, like actually write in the notes. There's space for it in the notes. Do you need to put in your homework document? It's only the textbook questions where there's no space to write it in your notes. You're asked to do it somewhere else. Those are the ones that would go in your homework document. Okay, so I want to talk a little bit here about how antibiotic resistance works, okay? And it's evolution. Short answer, evolution. But I want to give you a more detailed explanation here of how you get a antibiotic-resistant organism. And the long and the short of it here is that we use antibiotics too much. We're going to talk about selective pressures a lot more in the evolution unit, but this is an example of a selective pressure where we are constantly applying a selective pressure, and if you do it for long enough, you're going to select the bacteria that are resistant to antibiotics. And then those are the only ones that are going to survive. That's the basics of it. That's how evolution works. So for example, if you have a population of bacteria, and there's going to be variety in every population, just like there is in this one, some of them are killed by a particular antibiotic. I'm going to use erythromycin. It is a common antibiotic that's used in humans. And by the way, in animals for domesticated animals like cows and sheep and pigs and whatever. And some are always going to be killed by it. Often it's a very large percentage are killed by erythromycin bacteria. So it might be like 99% or 98%. Then you have some of the bacteria that are a little bit resistant, but they're often killed. They're not always killed, but they're often killed. And then you get the resistant erythromycin. There are going to be individuals that just by chance are more resistant to a particular antibiotic, erythromycin in this particular case. Okay? So we got this diversity here. There's a whole bunch, and you'll notice that most of them are red here. Those are the ones that are obviously killed. When you take an antibiotic, you're definitely going to kill the ones that are susceptible to erythromycin. So what ends up happening is that you get a new population of bacteria afterwards that is a couple resistant ones. Maybe it looks like this. Okay? Ideally, if you take the full course of your antibiotics, let's say that you are being prescribed erythromycin because you have strep throat. Okay? So you've been prescribed erythromycin. your immune system should, hopefully, kill those remaining cells, whatever's left over after you take the antibiotic. And that's assuming that you take the full course. And that is why you always must take the full course. Even if you feel better, you take the full course because you want to reduce the population to a very, very small population that your immune system is definitely going to remove. If you don't take the full course, you may end up leaving a fairly sizable population behind, and then your immune system may not be able to kill the population that's remaining after you take the antibiotics, and then you are definitely going to get a resurgence of your illness. In this case, it'll be much more resistant, and I'm going to show you why in a second, to the antibiotics. So you always want to take the full course. That's the first thing. Then, when this population regrows, let's say that your immune system is not able to kill the remaining bacteria. What happens is that when it regrows, the population looks like this. So what you have at the end, after you've done this erythromycin treatment, is that you've regrown a population, but now most of them have the particular DNA that makes it resistant to erythromycin. If you try and treat this infection with erythromycin, you will notice that it should be way, way less effective, because now most of the bacteria are not going to be killed by the erythromycin. Maybe some of them will, and hopefully that's enough to allow your immune system to fight off the infection, but it very well may not. And this is a bacteria or a antibiotic resistant bacteria at this point. You can now treat this bacteria with a different antibiotic. Okay, this is what we do in real life. So we give it another antibiotic, which is fine, but the same problem can exist if you give it another antibiotic, which is that you're going to eliminate the ones that are susceptible to that antibiotic, but you're going to leave behind the ones that are resistant to that antibiotic. And so it is possible to continuously do this and end up with a bacteria at the end that is resistant to all known antibiotics. There aren't that many. You can only make antibiotics so strong before you start killing the host. You start killing the people you're giving the antibiotic to. So there is a maximum strength here. And once you do that, you get what is called a superbug. A superbug is a bacterial strain that is resistant to many, if not all, antibiotics. And that's bad. That's bad. We've done that a lot. So we're getting into a bad area here where we've overused antibiotics. And by the way, when your immune system kills off this infection, it removes all of them. So if you don't require antibiotics for a bacterial infection, your body will just remove all of them, and you don't get this happening. So one of the problems here is that we're prescribing antibiotics often for nothing, for reasons that you do not require antibiotics. That is one of the biggest problems, is that we're prescribing them constantly for things that don't require them. Now, I must say that doctors are now trained to not do this, at least in North America, but there are lots of places in the world where nobody cares. But doctors are trained here not to do this. But there's a lot of pressure to do it. You know, a mom comes in with her kid, her baby or whatever, and says, oh, he's got an ear infection or whatever. And like the guidance is, we don't give antibiotics for this ear infection unless it's this severe. And there are certain things that they need to have before you prescribe antibiotics. But that mom doesn't care about that. They want antibiotics for their baby, and they want them now. And, you know, a lot of doctors are just going to say, fine, here, just take these antibiotics. What's the harm? And the problem is, this is the harm. There is a very good reason not to do that. And so I would say most doctors are pretty good about this now, although there are plenty that still break these rules all the time. However, this is a huge, huge problem. I'm going to give you an example. There are lots of STIs, chlamydia, syphilis, that we have fantastic antibiotic treatments for. Essentially, for many years, 50, 60 years, we have been able to cure these completely. No problem. You would go to the doctor if you had chlamydia, and we can cure your chlamydia, 100%. However, we've done this a lot. And now there are strains of chlamydia that are antibiotic resistant strains of chlamydia. So that you can get chlamydia now that is untreatable. There is no way to remove it with antibiotics. It's superbug chlamydia. That sucks. That sucks. I mean, we did that. We created that situation. We absolutely did not need to do that by just being so flagrant with our use of antibiotics. And the same problem exists with animals. For example, cows. No, we don't do this in Canada. It's not legal to do this in Canada. But again, there's lots of places in the world that do this, that just always give their animals antibiotics. They're just always receiving antibiotics constantly. And that's just to keep the herd healthy all the time. Does some of that get into the milk? Maybe. It's possible. It's probably likely that some of it is. Which again has a knockdown effect for humans, because then humans are constantly consuming it as well. This is a problem in the US, by the way. But I don't know why they do this. They really should know better. It is more profitable for farmers because you get less sick animals. There's less animals that are less sick. However, you get superbugs in your herd. It is getting more and more common to have infections in cattle that just are not treatable by antibiotics. I mean, guys, this stuff bothers me so much. It's an obvious solution. It is more expensive to go the other route and not treat these things continuously. However, in the long term, it's like essential to do this. Anyway, I don't want to get into this too much, but these strains are out there. It's important for you to know this, because when you go to the doctor and they tell you there is no antibiotic for this, you must accept that. Because we are all part of the problem here, in that we request antibiotics for things that make no sense. Now, I should mention that antibiotics also only work for bacteria, and the vast majority of stuff that you go to the doctor for is viral. And so there is no way to treat those illnesses most of the time. Rest, fluids, but there's no drugs that you take for viral infections for most of the time. And there's some exceptions like HIV, but a lot of viral infections there really is no medication for. So this is something we just have to accept as humans, that like there's no magic pill for a lot of the stuff that you get. Okay, what's the science behind anti-symptomatic symptoms? Asymptomatic? Oh, sorry, you corrected yourself. Asymptomatic symptoms. Are you talking about asymptomatic transmission of an illness, Kim? Like, for example, asymptomatic COVID, which people talk about all the time? Or like a typhoid Mary situation, where you have somebody who's carrying typhoid Mary, who is not showing any symptoms of typhoid but is fully contagious. I'm going to assume that that's what you mean. I don't know what the delay is on this. Okay. So basically how that works is the infection is there. There is a population that is significant enough that it can be transmitted to another person. However, your immune system is doing a fairly good job at managing that population within your body. However, it is not eliminating that population from your body. So for whatever reason, your immune system is simply keeping the illness in check without actually removing it. Or in the case of some asymptomatic infections, for example, with COVID, you may be fighting off the infection and it is extremely minor. So you've managed to keep the population fairly low, which by the way, generally means that you are less infectious. You're not as infectious when you're asymptomatic as you are when you're like sneezing and coughing large droplets of whatever infectious agent into the air. However, you are still infectious. You still, depending on the type of illness, you may have a high enough population to actually transmit it to others. That's the problem, especially with the new strains of COVID, is that it's so infectious. It requires such a small initial population of bacteria, or virus in that case of COVID, that even though you've managed to keep it in check and you're not experiencing any symptoms, you still are able to pass on enough virus particle to somebody else just through talking or maybe direct contact. that you could potentially give them COVID anyway, or whatever you name the illness. So it is possible to transmit. There are lots of illnesses, by the way, that you can transmit asymptomatically, but it is less likely. Usually because the symptoms themselves are a means of transmission. Coughing is a means of transmission for an illness. It's literally encapsulating and firing particles of that illness at another human, which is, by the way, a reproductive strategy for viruses and bacteria. If you want to get into another person, make that person cough. I mean, it's a great evolutionary trait to have as a bacteria or virus if you want to get into another human and keep replicating. So that's why those things evolve in the first place, because they're great at carrying on a particular strain. Okay, I'm going to stop talking about this now, that particular topic anyway, and I'm going to very quickly discuss archaea. Archaea are not as common as bacteria. However, there are still many different species. I'm going to go over some of them now. And by the way, I'm not talking about specific species here, but I am talking about groupings of species. These are broad groupings of archaea, not specific species. And often they're categorized by their type of extreme environment coping mechanism. Archaea are cool in that they are able to survive in these really dramatic environments that are very difficult for a lot of living things to cope with. So, for example, anaerobes, anaerobic environments are, in general, difficult for bacteria, or I should say prokaryotes, to survive. Most of them have anaerobic metabolism. Methanogens do not. They live in your gut, although there are other places where methanogens live, but they produce methane as part of the metabolism. So I'm sure you've experienced gas before. When you have gas, a lot of it is because of the class of archaea that live in your gut, and they produce methane. So those are called methanogens. Halophiles are archaea that are able to live in very high salt environments, saline environments. Typically, prokaryotes are killed by large concentrations of salt because the salt pulls water out of the microorganism. It causes it to dry up, to shrivel up and die if it's a very high salt concentration. But halophiles, you can see them growing on like salt lakes and like salt plains, salt flats, places where they're basically completely devoid of life, except for halophiles. They'll pretty much be the only thing that lives there. So that's kind of an interesting adaptation. If you're interested, by the way, in the physiology, the biochemistry of how they adapt, there are really cool courses in these things in university if you're interested. Because it's a fascinating method that they use to cope with these. I'm not going to get into it here because it's way beyond the scope of this course. Extreme thermophiles are archaea that live in very, very hot temperatures. And not only live in very hot temperatures, they require very hot temperatures. They can live in water that is up to 114 degrees Celsius. That's far above boiling, mostly in thermal springs. But they live on the sides of volcanoes, in thermal vents, and thermal pools, in geysers. If you've ever been to Yellowstone or something like that, you've seen... Oh, actually, I've got to show you a picture. There's some absolutely beautiful ones in Yellowstone. Maybe you guys have heard of the Grand Prismatic Spring. I went here a few years ago, and it was absolutely beautiful. It looks fake. Like, it looks like it wouldn't look like that in real life. But it is, I guarantee you, it is absolutely this vibrant in real life. So, there we go. That's a good picture of it right there. This is the Grand Prismatic Spring. There's a little walkway to the side there. And this is a thermal vent. So, Yellowstone Park is built on top of a giant volcano, supervolcano. And there are areas where thermal vents are allowing superheated water to get to the surface. This particular hot spring is called the Grand Prismatic Spring. It is boiling. And different types of extreme thermophiles live at different temperatures. So the bright blue ones that live in the center are the hottest of the extreme thermophiles. So they live in the 100 to 114 degrees Celsius range, and so they grow there. And then the different colors are actually different species here of extreme thermophiles. As you move towards the outside, the water is cooler and cooler, and different extreme thermophiles, or archaea, live at different temperature ranges. So as you get to cooler and cooler temperatures, you get different colored extreme thermophiles, which is cool. If you ever get a chance to go to Yellowstone, this isn't the only one. There's a number of these thermal pools, and they're absolutely beautiful. Okay, and then you got the opposite of extreme thermophiles, which are psychrophiles. What a cool name. Psychrophiles live and reproduce in very cold environments. For example, they can live and thrive in the high Arctic, -40, -60. And they reproduce actually fairly well at temperatures below freezing, which for most organisms, especially microorganisms, they do not survive at that temperature. Well, they survive, but they don't reproduce. Their metabolism is halted. But psychrophiles are able to reproduce even at very cold temperatures. Now, these are not pathogens, so if you wouldn't have these in your food anyway. But they could potentially grow in your food if you put them in the freezer. But they're not pathogens. They're not pathogenic. So you don't have to worry about getting sick from psychrophiles growing in your freezer. That would be first time, probably. Anyway, those are the analysis questions that go along with the prokaryote section. They're also listed on Brightspace. So those are the ones that would go in your book, and we'll come back to them. Okay, I'm going to go a little bit faster here because I've been stopping for questions and stuff, and I got a little bit behind, and I want to give you guys time to work on the stuff that you need to work on. So I'm very quickly going to go through viruses. So what makes viruses cool? Well, let's talk about what living things do. I just made a quick list. Okay, these are just what living things do. All living things, the definition of a living thing. And you probably would have talked about this in grade 8. This is in the grade 8 curriculum. So they respond to stimuli. Maybe if there's increased light or heat, or they do things. They might move towards the heat or move towards the light or change their metabolism or whatever. They can respond to external stimuli from the environment. They eat, so they have some type of metabolism where they take in food. They could produce it from light, but either way, they are producing some type of metabolite. They adapt, so they are able to change their biochemistry to their external environment, depending on the conditions. There's lots of ways even microorganisms adapt to their environment. They can reproduce. That one's key. You have to be able to make smaller versions of yourself. They make waste products at the end of their metabolism, and they grow. So they don't start and stop all at the same size. Okay, so those are in general considered to be the properties of living things. If a thing does this, we say it's alive. Some people would add to this list that living things are made of cells. That's also true, but I'm not sure that you'd necessarily include it in this list. But it is also true that generally living things are made of cells. And by generally, I mean absolutely. All living things are made of cells that we're aware of. Now let's look at things that viruses do. Okay? Do they respond to stimuli? No, not at all. They are a molecule. Do they eat? Nope. No metabolism whatsoever. Do they have any type of environmental adaptation? No, not at all. They cannot move or adapt to their environment in any way. Do they reproduce on their own? No, they don't do anything on their own. Do they produce waste products? Nope, not a one. Do they grow? Nope, they're always the same molecule and they're always exactly the same size. So if we look at this list, we don't check any of the, hey, I'm a living thing boxes. Which is why we generally don't consider viruses to be living, because they don't do any of the living things. However, they do act sort of like living things in that they can do these if another organism does those things for it. So if it can go into a cell and it can tell that cell to do those things for it. Not all of them, but some of these things for it. So they live in this weird gray area. We don't consider them living, but they do kind of achieve some of these things by asking another organism to do those things for it. So we already talked about their size. They're very small. They're non-living for the reasons we just stated. And basically all they are is a piece of DNA, some genetic material. Whoops, a daisy. Screen here. Some genetic material surrounded by a capsule made of proteins. And that capsule is called a capsid. That's all. So a little piece of RNA or a little piece of DNA. So when I say genetic material, I mean RNA or DNA. So DNA would be the type of genetic material that you use to pass on information to your offspring. Or RNA, which is single-stranded. Those are called retroviruses because they get the host organism to build a piece of DNA from their RNA and then incorporate it into their own DNA. But anyway, they have no cytoplasm, so there's no goo. Although their capsid may contain some enzymes that allow it to function within the cell, but it's not strictly cytoplasm that's inside. They're tiny, already mentioned, less than 1 micrometer in diameter. And in general, you could fit thousands of these inside one of your cells. Depending on the cell size, you can fit hundreds of thousands of them inside of your cells. They don't grow or reproduce, as we just mentioned. They don't use energy or produce energy. They don't make waste products. So basically, you can just think of them as a package of DNA or a package of RNA that has instructions for making copies. Okay? Mostly copies of themselves. That's what it is. It's a floating instruction booklet that tells other things how to make copies of it. And that's it. Cool idea. That's how it works. So you're probably already aware, you would have talked about this in grade 10, this idea that your cells make proteins from DNA. They read their DNA and they use ribosomes to make proteins. We're going to talk about this in more detail actually in the genetics unit. It's the beginning of the genetics unit, which is next. So I'm not going to go into detail here on how that works, but that is what you do. Your cells read your DNA and then they make copies, or they make proteins based on whatever it tells it to make in the DNA. So what the viruses are doing is just giving your cells an extra piece of DNA or RNA. Again, you can incorporate either of those. And just saying, hey, here's some instructions. Make this instead. And if your cells have no defense mechanism, they often don't, they just say, okay, I'll just make this a whole bunch of times. And so it makes a whole bunch of viruses. Then the cell lyses, often. It explodes because it's full of virus particles. And then those go out and attach themselves to new cells. And then the new cells do the same thing. They take the instructions in and make a million and then explode. So obviously that can snowball into a larger and larger population of viruses. And that's a viral infection, in short. Okay, COVID works like that. Virtually every virus infection works like that, although they are usually targeted to specific types of cells in specific organisms. They have specific hosts. So, as mentioned down here, most viruses only have a single host species. Some have crossovers. Some viruses can infect all mammals, for example. However, most of them are single host species, have a single host species. HIV is an example of a virus that is probably specific to humans, although it may be able to infect primates. But outside of primates, there is it's a different virus. So for example, there is a feline version of HIV, but it is only for felines. It cannot be spread to other species, barring some kind of unusual interspecies transfer event, which can occur. And that is probably what happened with COVID, by the way. It was probably transmitted from bats. So there was a single incidence where a certain population of virus, due to natural selection and random mutation, was able to cross over into another species. That's probably where COVID came from. But anyway, in general, they are limited to a specific host. Although there are some that infect many species. Rabies is a good example. So rabies is one that affects all mammals, to my knowledge. So pretty much any warm-blooded organism, any mammal, can pass rabies on. Mice, dogs, cats, rats, bats, whales, I guess. I mean, you could. I've never heard of a whale having rabies, but they're probably can be infected by rabies. We only ever talk about certain target species for rabies where it's more common. Bats are a good example of a species that often carries rabies. Rabies is fairly common in bat populations. However, you could get rabies from almost anything that would potentially bite you in the wild. So that's why you get a rabies shot if you're ever bitten by a wild animal, or even if you have just contact with the saliva of a wild animal. A bat has landed in your hair, and has made physical contact with you. You need to get a rabies shot for that, believe it or not. Just as a heads up, if you ever have contact with wild animals, get a rabies shot. It's not that bad. I know plenty of people that have had to have a rabies shot. It's not. It's a little bit on the painful side because you have to get multiple injections, but it's really not that bad. And then you don't get rabies. By the way, there's no cure for rabies. Once you show symptoms, it is 100% fatal. It kills everybody who becomes symptomatic. That's scary. 100% fatal. You don't hear about that very often. So you definitely going to want to get that rabies shot. Anyway, and then you can also find some viruses that infect bacteria. They're called bacteriophages, I already mentioned. Okay, so as I mentioned here, are they alive? No. Sometimes I get you guys to do these questions, but they're pretty straightforward. I'm not going to make you answer that. They're non-living. They don't have any of the characteristics of living things, as we mentioned. They can't produce, can't reproduce, they can't consume nutrients, they don't produce waste, they don't respond to stimuli without a host. And I guess that's an important point here, is that they kind of do those things, but a host has to do them. So they don't do them on their own. Maybe to go back to your question from before, Trenton, I would say that they're like pseudo-living things. They do living things, but through something else. I don't know how you would apply that, but... And where do they come from? Interesting story. Probably, this is our best hypothesis, although obviously we have no evidence. I mentioned this previously. There's no fossil evidence for viruses. But they probably originated as a small parasitic cell that lost their ability to reproduce outside of the host. Probably. I mean, that's a good guess at a mechanism that could have existed for the origin of viruses. Or it could have been a piece of DNA or RNA that escaped from an early cell and was able to be taken in for whatever reason into another host cell, replicated, and then passed on to an additional host. So, and it's also possible that they evolved multiple times over all of biological history. It's not that that event could have only occurred once. So when did it happen? Nobody knows. It could have been fairly recently, like in the last 50,000 years, or maybe viruses are as old as life. We really just, it's a complete guess. Although I do think there is some good evidence to suggest that viruses aided in speeding up the evolutionary process by creating random mutation. And so we're probably around for a long time. But again, we don't have direct evidence. Okay, why do we care? Why do we care about viruses? Well, mostly we care about viruses because they cause human disease. Many. Oftentimes, a lot of the illnesses that you get over your lifetime will be viral. Every cold you've ever had was probably viral. There are some bacterial infections that mimic the cold, but to be perfectly honest, almost every cold you've had is probably a viral infection, which means that there is no treatment. Chicken pox, AIDS, cholera, rabies, which you're probably haven't had those ones, although you may have. They can be potentially deadly. So there are viral infections that are often deadly. Some of them are fairly innocuous, like chickenpox, although you can die from chickenpox. It's possible, but it's quite rare. COVID is one of those ones that fits in a weird middle ground. So COVID is relatively mild infection for most people. But the problem is around 2 to 3% of the population will experience severe illness with COVID. And that's a high number. It doesn't sound like a lot to say 3% of the population has severe symptoms. But the amount of medical care that's required for that 2 to 3% is extremely high. I mean, it requires intubation and like extensive medical treatment. Often the symptoms last for a very long time, which is one of the reasons why it's so dangerous, because it places such an extreme strain on the healthcare system. If 100,000 people get COVID, that's thousands of people in intensive care units. That's a really, really high population of people that we have to treat with a lot of resources. And obviously, you don't want to be that person who goes to the hospital and they say, we don't actually have any space to give you that life-saving medical treatment. I mean, that's scary, right? We expect that life-saving medical treatment, which is one of the reasons that the lockdowns and things like that exist. It's to ensure that the course of transmission for a particular illness, COVID in this particular case, is slow enough and it grows slow enough that we actually are able to provide treatment for everybody. Because it's a very scary situation when we're not able to provide that treatment. So anyway, that's the reasoning behind a lot of the control measures, is to ensure that we actually can treat people that are ill. Speaking of that, I'm going to get you guys to do a little bit of a terminology search on your own. What am I doing for time here? Holy cow, I've ever been talking long. I think I'm going to give you guys some time after we come back and take attendance after the break. I'm going to have to give you some extra time to just work on this before we do the last section, because I've given you no time to do this stuff, which is not cool. This how are we doing for tomorrow? Let me check. Maybe I can bump the last section to tomorrow, actually. I've been talking an absolute ton today. What's on tomorrow's docket? Yeah, I'm going to do that. I'm actually going to bump. That's exactly what I'm going to do. So on the day 3 stuff on here, all of the protist stuff that's at the bottom here, we're going to bump that to tomorrow. So don't do any of the learning block 2 stuff today. That's the protist stuff. I'm going to bump the entire thing, because otherwise that's a ridiculous amount. That's too much stuff to ask you to do in one day. So tomorrow is actually a relatively short day, and we'll be able to fit that in much better tomorrow. Okay, so just as a heads up here, don't do the protist stuff. Anyway, I'm going to get you to do this research on your own. I want you to look into what is an epidemic. What does it mean if something is endemic? I just added this in afterwards, but I want you to look that up as well. What does it mean if something is endemic? Hint, hint. I have a strong suspicion that COVID will become endemic, not eliminated, as I'm sure some people are hoping for, but I think it's unlikely. What is the current status of the coronavirus pandemic? Is it pandemic? Is it an epidemic? Is it endemic? What is it? What do we call it? And I'm going to get you just to research that on your own. That won't take you long to find out. There's about a bajillion sources you can look up for that. So the last thing I want to mention about viruses here. I'm going to talk about vaccination for a second too. But I want to mention just what do they look like? What are the characteristics of a virus? The one that everybody always asks about is it lunch now? It is in 2 minutes, Trenton. And you know what? I'm actually going to come back and just finish off this page when we come back at 11:05. And let's take a break. I'm going to stop talking. I probably need to drink some water too. I've been talking way too much. Guys, it's been a pleasure. Enjoy your lunch, and I will see you guys at 11:05 for attendance. Okay, cool. So yeah, again, Evan, if you haven't. Oh, you got me. You got me. I got your email. Good. All right. So just a heads up, some people sent me some emails over the break. I will get to those. I usually take a quick pause to have a snack and hug my baby and take a moment. So I don't always get to those during the break. Sometimes I do, but I will definitely get to them with my earliest opportunity. Okay, let's quickly finish off our discussion about viruses, and then I'm going to give you guys an opportunity to get to work. Remember that if you have questions from the homework, you can email. It's fine, but it is probably better if you drop in to the Learning Block 3 question period, because I'll be able to give you the best answer if you do that. Over email, sometimes it's difficult to provide quality explanations, but it's a lot easier to do if I can, write on my iPad or whatever and discuss things with you face to face, or at least verbally. Anyway, so we were talking about characteristics of viruses, what they actually look like. Most viruses have some kind of protein coat around the outside. So that would be like this part, the bumpy part on this particular. This is a tobacco mosaic virus, which is specific, obviously, to tobacco. But there's usually some form of capsid around the outside. So on this adenovirus, it's this area. I probably should use a different color than that. It's hard to see. Maybe this. Oh, it doesn't work either. What shows up on green? Blue? Not at all. You can't see it. Oh. Every once in a while, this thing decides that it's not going to show up. Sorry about that. Thank you for letting me know. Please let me know. There we go. So, as I was saying, on the outside of a virus is usually a capsid of some sort. So it is like a shell made of proteins on the outside. It protects the important DNA that's in the middle. And it's different depending on the type of virus. And the shapes of viruses can be all different kinds of shapes. This is a tobacco mosaic virus. Again, they're specific to specific hosts. So this infects a tobacco plant, obviously. This is an adenovirus. There are many, many different types of adenovirus that have this sort of geometric shape to them. There are human adenoviruses and adenoviruses that attack all kinds of different organisms. It's based on shape. But they all have this sort of like protein coat on the outside. They also often have some kind of envelope outside of their capsid. So the capsid is what's enclosing the DNA or RNA. So again, it can be a DNA or RNA. If you're not 100% sure of the difference, DNA has two backbones, and then it has the various letter combinations, the various code going across the center. You probably learned this last year. You might not have. Whereas RNA is only half of that. Oops. It's half of the backbone. So this would be DNA, and this is RNA. RNA is also used in eukaryotic cells like yours to send a message from the nucleus to the ribosome to tell it how to build a specific protein. That we're going to go over that in the genetics unit, so you don't need to know about that right now. But just be aware that the actual heritable information, the information that is containing of the instructions for the virus, can either be DNA, which is like the full double-sided helix, or it can be a single-sided RNA molecule, depending on the type of virus. And then usually on the outside of the capsid is what's called an envelope. Not every virus has an envelope around the outside, but a good example of one that does is the coronavirus, which we'll be discussing shortly. So where the purpose of the envelope is essentially to create a envelope, it's got a perfect name, that goes around the outside of the virus that mimics the outside of the cell of the host. So, for example, if this was a virus that would be infecting humans, the envelope going around it would be what looks like a cell membrane, a lipid bilayer that goes around the outside and has a number of proteins sticking off of it. And those proteins are the same proteins that you would find on the outside of your own cells. So when this particular shape comes into contact with the outside of one of your cells, so this is like floating around, and it comes into contact with the outside of one of your cells will think, well, that's just a piece of my cell membrane. And often it will incorporate that cell membrane into itself and pull whatever the thing is in the center of the envelope into the inside of the cell. So it will allow the virus to merge with the surrounding cell. This also makes the virus very difficult to detect by your immune system, in multicellular organisms anyway, because it is disguised as one of your cells. It looks like and has the surface proteins of one of your own cells. It doesn't have anything that sticks out that says, oh, this is dangerous, this is a virus, immediately. So it's not immediately obvious. However, your T cells will learn, because this is not perfect, there are going to be proteins on the envelope that are virus-specific proteins. So you'll get these little flags that are sticking up that are virus-specific. And maybe your body doesn't identify them immediately, your white blood cells don't identify them immediately, but eventually they learn how to identify those shapes that don't look quite right and essentially put little flags on them that say, hey, this is a bad guy. I call them bad guy flags. They're also called antibodies. But it will learn how to flag those inappropriate proteins. And that's how you become immune to a particular virus. And you learn how to build these flags for those specific structures, and you're able to very quickly mark them when they make their way into your body so that you don't get a full-blown infection of whatever the particular illness is. That's how immunity works. So if you've already had COVID, chances are you've mounted a pretty good immune response, and you've learned how to build some of these flags, which are going to be now why am I not able to do this? Yeah, which are now floating around inside your blood, and then when they come across the proteins for that particular virus, they'll be able to attach and say, whoa, bad guy flag. This is a bad guy. Let's destroy this immediately. Don't let it get into any of our cells. It's pretty rapid. Now, that immunity usually dwindles over time. You stop producing less and less of the specific flags over time, and that's why a lot of vaccines require a booster shot. We'll talk about how vaccines work in a second. But often this immunity doesn't last for your entire life, so it does tend to dwindle over time. But there are some early indicators that people who get COVID do have some long-term antibody protection. How long, we're not 100% sure. It hasn't been around that long. So and people can get reinfected. It is possible. Sometimes when you get infected the first time, you don't mount a very strong immune response, and you just don't create a good force of flags. So maybe you just don't make very many of these flags, and it doesn't give you complete immunity, and then you may get infected again. It does happen. Or you have a depressed immune system, and your immune cells are not doing a great job at making flags, and then you might get infected again. So, but there is often a pretty strong immunity that is conferred from being infected. Would variants be chemically similar? So now that's the good question. Michaela, fantastic question. That is the question that we don't have a great answer for. Some of the variants are similar enough. And when we're talking about differences between variants, what we're really talking about here is differences between the flags. So there are more and more varied flags. The variants have slightly different flags. Not all of them are different. Definitely not all of them are different. Some of them are different. So sometimes the flags are not significantly different in a new strain or a variant. The variant is really a strain, but that the immunity that's conferred to one strain will be the same immunity because essentially the flags still fit. Your little immunity flags will still stick, or maybe the same protein is there and they'll stick to that protein. But not always. So sometimes a new variant arrives and you don't have a good compatibility between these antigens. They're called these shapes, by the way, that are on the surface of. Do I write that on here anywhere? Yeah, I do. I've written it over here. So the little shapes that you see on the outside of the proteins here are called antigens. Or on the outside of the virus, rather, are called antigens. So that's what you're really building your flags to the shapes, to the antigens. Yeah, Kim, we do that all the time. We use viruses as vectors to intentionally put specific DNA inside of specific cells. That's how gene therapy works. Gene therapy is sort of still in its infancy. It's only been around for maybe 10 years. And we don't really use it for a lot of things yet. But if your cells, for example, don't have a gene that is needed to produce, I don't know, a really important protein, let's say insulin or whatever, there's a million proteins you could choose, you could potentially use a virus to inject that new gene into those cells. And so you'd be manipulating or using an already existing virus to help you inject some new DNA into a cell and potentially produce a new protein. And we do that. We use viruses as vectors for that. Complex process, there are some potential side effects of doing stuff like that. Well, we used to spend actually a bunch of time in this course on that, but we kind of shifted our focus away from viruses since they're not technically alive. It's not really in the curriculum, so... but it is fascinating. Yes, viruses can be manipulated by scientists to do all kinds of things. Really interesting, burgeoning area of virology is using viruses to kill bacteria. I mentioned previously this problem that we have with bacterial immunity to antibiotics. However, if you can design a virus to kill a bacteria, a bacteriophage, kind of like this bacteriophage here, you may be able to use that as an antibiotic, inject that into a person, and have the virus kill the bacteria. It wouldn't inject it into your cells because they're not compatible with the virus. And that could be potentially a new treatment. That is an area of examination right now that's very big in science, is this idea of using viruses as antibiotics. Because they generally do not become less effective over time, the way that antibiotics typically do. So, I mean, that would be amazing if we could figure out a way to use viruses to treat bacterial infections. Would an immunity to corona help against other respiratory viruses that could develop in the future? Maybe. So sometimes there are common antigens between different species of virus, but generally not. There is a lot of variety in the shapes that exist. Colors probably don't do it justice here, because the variety of shapes that you can find on the outside of virus particles is, almost infinite. So there's not a ton of crossover between viral species, and so there's no such thing as like a general immunity, where that you have a coronavirus infection and it confers immunity to some other random virus. There is a little bit of crossover in antigens, but it's very, very small. So that is not typically a thing that exists. There are vaccines. I'm going to talk about vaccines in a second, but it essentially protects you from a particular set of antigens that do have some crossover between species, but they're designed specifically that way to have crossover. So I would say the answer to your question is generally no. It's not a broad immunity to a number of different viruses. And I'll give you an example. The flu. The flu usually mutates significantly from year to year. So the flu vaccine that you get every year is a completely different vaccine. Well, sometimes it has variants that are carried over from year to year, but often it doesn't. And the flu vaccine from 2 years ago does not protect you against this year's flu. because you basically need to re-engineer the vaccine every single year in order to keep it effective for the new variants that exist of the flu. I think that there is a very strong possibility that the COVID vaccine will work this way, that it will protect against this year's strain of COVID, but not necessarily future strains. Although there is some evidence to show that the variants of concern that people are talking about right now are at least semi-protective, or at least the current vaccines are at least semi-protective for those variants. Although the data is still very lacking in that area to know that for certainty. But there's some early indicators that the vaccine does protect against a number of the variants. And when I say vaccine, I realize that there are several different vaccines, but they all, to my knowledge, seem, at least certainly the ones that are used here, seem to confer at least some level of protection to the other variants as well. Although, again, that area of science is an open question still, so we don't have a lot of great evidence for that. I'm hopeful. Once we establish a process for creating a good COVID vaccine, which we actually seem to have done already, producing a new vaccine every year and distributing a new vaccine every year is actually not that difficult. We do it with the flu already, although we don't vaccinate on such a large scale with the flu, although we probably should. But we could do that with fairly low trouble. So the main issue with this vaccine is that these are new vaccines. They're using new methods, like the mRNA vaccine is basically a new method. It's never been used on a large scale before. So in order to make sure that those vaccines are safe, a lot has to go into trialing those vaccines to make sure that they are not going to end up causing health problems for people in the future. Once you've done that groundwork and shown that that method is safe, creating a new vaccine with a new variant and then having that apply for a subsequent year really doesn't take that long. It's actually quite a fast process. Making the vaccine is quite a fast process, and distribution, once you have the network in place, is actually quite fast as well. So I don't think it's out of the realm of possibility that we would have yearly vaccinations. And people are like, oh, every year, that sounds crazy. But we already do that with the flu. I mean, I get my flu vaccine every year already. So it's not like it's a crazy idea to have yearly vaccinations for COVID. Again, this is not like a guarantee, but I am strongly suspicious that may be the way that this pans out. No one talks about it because, of course, it's expensive and it would entail significantly more resources being invested in this, which people don't want to think about right now. But I think that there's a high chance that that's probably the case, but we'll see. We'll see. Obviously, that we're not there at this point. Sorry. So like bacteria, not all viruses are bad? Absolutely. Most viruses don't infect humans, so they're not bad for us. They don't have the right antigens to be able to penetrate into our cells. They don't have the right coating envelope on the outside to penetrate into our cells. So when you say it's not bad, I mean, not bad for us. Certainly other species, like species we raise for food, there are susceptible to different types of viruses. There are lots of plant viruses, so food crops can be susceptible to virus infection as well. And things in the natural environment as well, various ecosystems. They're usually held in check by, evolution at a natural level, as long as they're not engineered, because of the variety that exists in nature, because of the genetic variety. Of course, if we eliminate the genetic variability in nature by removing such a large amount of species and populations from the natural world, we make them more susceptible to virus attack. But assuming a population is healthy and an ecosystem is healthy, they're generally fairly resistant to viral attack. So yes, they're not all bad. They're not all bad for humans anyway. And we can potentially use them as a tool, the bacteriophage. There are not so many helpful viruses. So when we talk about bacteria doing a lot of helpful roles, we can use them as a vector for experimentation and for medical treatments, but there aren't like a lot of viruses. Oh, I shouldn't say that, because they do keep certain bacterial populations in check in nature. They prevent any particular bacteria from multiplying out of control. Often bacteriophages will prevent that from occurring in the natural environment. And so I guess there are good versions of viruses from our perspective as well. They're like environmental regulators. But they just don't serve as many useful roles as bacteria do. I'll put it that way. Okay, so what have I written down here? Essentially that viruses have all different kinds of structures. There's many. You see some of them above. They vary in structure, but they all have RNA or DNA surrounded by a capsid or an envelope, or capsid and an envelope, depending on the type of virus. Not all of them have both. So some of them just have capsids and no envelope. Bacteriophages have no envelope. They also look like really cool aliens. Or at least I think they do. The envelope is usually created when the virus particle is created and it leaves the cell that created it, which is kind of cool. So then it kind of disguises itself as one of your own cells. Leaves your host cell, or the host cell of whatever organism. And then it is able to penetrate into another host cell, or maybe leave the host and penetrate into another organism with similar envelope. So it would be same species usually. So I kind of discussed this already, but I really briefly want to go over what a vaccination is. I kind of mentioned this already, but what a vaccination does is it gives the host, whatever organism, humans are not the only people that can be vaccinated. You can vaccinate sheep or goats or whatever, any animal, plants. You can give vaccinations to plants. They have a form of immune system as well. Although they're different, obviously, they wouldn't be the same type of vaccination. But I mentioned that the bad guys, the viruses, and bacteria too, by the way. Bacteria have antigens on them as well. So vaccinations are not only for viruses. They work for bacteria as well. They have some shapes on the outside. I've done a better job below here at drawing the shapes. And that's how you recognize whatever that bad guy is, virus, bacteria, whatever it happens to be. It actually could be a parasite as well. So it could be a multicellular organism as well. But they're shapes. You can give those shapes to a healthy host like me or you, and just the shapes. You don't have to give them the whole virus. Now, you can give them the virus, a broken version of the virus, or a weakened version of the virus. That's one way to make a vaccine. Those are pretty unpopular. So those are called attenuated. Actually, no, wait, that's not what the word attenuated means. Attenuation is a way of making the immune system react stronger to the virus. Live attenuated viruses are ones where you give a living virus essentially to somebody, and then you let the immune system react to it. But it's like a version of the virus that has been weakened, so it doesn't give you illness. Like I said, those are not very popular. We don't use live attenuated vaccinations very often, but it is a strategy. The new way of making vaccines, the mRNA strategy, which is what the Pfizer vaccine is, if you guys have heard of this before, is so super cool. What they do is they look at the DNA of the virus. So they'll go through and read the DNA of the thing. Okay, they go through, read all the base pairs. And then they'll figure out, okay, this section right here makes this shape. They do this with computers, by the way. They computer model what that piece of DNA will make, what it builds, what shape it builds. And then from that, they will build the shapes. Just the shapes, just the antigens, not the whole virus. But they basically are using a computer to simulate what the shape is, what the antigen will be from the virus, and then pick some shapes that they think will be key shapes that will always be maintained in that virus, or is maintained as much as possible. When I say maintained, it means when the virus makes copies of itself, that shape is usually there. It's an important shape. The problem with making a vaccine is that they mutate so quickly. Their shapes change very quickly. And so that, like the flu virus, for example, where you have to make a new vaccine every year. And that's why there is no vaccine for everything. Some things mutate so fast that it's actually impossible to produce a good, viable vaccine that covers all the different shapes that are out there, or even a large percentage of the shapes that are out there. HIV is a perfect example of that. So HIV is extremely rapidly mutating. So fast, in fact, that if you have HIV, the HIV that you have when you start and the HIV that you have a year later may be different. It may be different enough that it wouldn't be the same antigens from year to year. And obviously that's very problematic. How do you build a picture of these shapes to make a vaccine if it's so different from year to year? And in which case, for HIV, it's been impossible so far. We haven't been able to find any domains, any places that are the same enough to build a good vaccine for it. And there's lots of viruses like that where development of a vaccine would be extremely difficult. Before we made the COVID vaccine, we weren't sure if that was true for COVID. It may have been, but lucky us, it doesn't appear to be. And so we do have quite an effective vaccine, actually, several of them that are made in different strategies, but they all appear to work fairly well. Can we use science to predict the mutations in shapes? From year to year, we can guess which will be the popular shapes. We can't necessarily guess where mutations will occur because they are generally random. So how they will change from year to year is it's random. So there's no way to make like a random predictive algorithm. Maybe it's not totally random, but it is as far as we know. So maybe if we figured out how those mutations happen, if there was some consistency to them, we'd be able to figure that out. But we don't have that methodology currently. However, So with the flu vaccine, for example, what we do is we look at what variants are out there for the flu, and then we predict, based on how much of those variants appear to be out there, how we should build the flu virus for the next year. Based on which variants are the most common, we're basically guessing which will be the popular versions of the flu. And we make the vaccine for what we think will be the popular versions. Usually we make a good guess, and the flu vaccine is fairly effective. Sometimes we make a terrible guess. So there have been years where we've made a guess for the flu vaccine that has been a terrible guess, and the flu vaccine has actually been very ineffective. But that is quite rare. Most of the time we make excellent guesses. We have a pretty good way of making guesses. And so in general, we make a good guess, and we get a very effective flu vaccine at the end of the year, or during the flu season. Because obviously you have to guess before the flu season to make your vaccine and have it ready for the flu season. So there is some guessing involved, and usually they're good guesses, but not always. Anyway, speaking of the mRNA, so we build these specific shapes, we give the specific shapes to people, and then your white blood cells, in this case they are T cells, are going to come and start building flags. So those flags are called antibodies. And we already talked about this. Whoopsie-daisy. We already talked about this idea. But your white blood cells are going to start building these flags, and that way, when that particular virus or bacteria shows up, hopefully you already have a system of flags to stick on them and inactivate them and potentially flag them for your immune cells to eat them or remove them. This is the great victory of science. I don't think people realize how important vaccination actually is. We have been able to eliminate some of the worst illnesses in human history, things that have been devastating humanity sort of since the dawn of time. I'll use smallpox as an example, which we've eliminated, by the way. As far as I know, smallpox doesn't exist anymore because of a vigorous vaccination campaign worldwide. But smallpox is often fatal. Oh, I don't know if you want to see this, guys. It's not nice. But it causes really, really terrible pox. I mean, it's very similar to chicken pox, but it's so aggressive, and it covers so much of the surface area of your body, and it is often fatal. It's really a scourge on humanity, but it doesn't exist anymore. Which is because of an aggressive vaccination campaign. I mean, it used to be so common. We have so many success stories like that with vaccines. I mean, vaccines are amazing. I think sort of in modern times, because we've eliminated so many really terrible illnesses through vaccination, people have like lost their trust in vaccination. I don't know what happened with that. I really don't get how that occurred. Maybe there's some kind of pushback against science in general. I'm not really sure exactly what happened, but anyway, vaccines are a miracle, guys. they're really amazing. If you have the opportunity to get vaccinated right now, no COVID vaccine is approved for young people yet, but I anticipate it to be. I mean, that stuff is coming. It's being tested on young people right now. Almost certainly it will be approved in the near future. So, I mean, get vaccinated if you have the opportunity. Some people are immune compromised and vaccinations do not work for them. And that's one of the reasons why it's so important to get herd immunity, to get so many people vaccinated in the population that it's just unlikely that anyone who's unvaccinated will get ill. And we need to do that for vulnerable populations that cannot be vaccinated. For example, for people that have weak immune systems. So anyway, something to consider. And I don't understand how people are not supporting vaccination, but you definitely should support it. The side effects that you get from vaccination, which are like really rare allergic reactions and things like that, 1 in a million, 1 in 10 million. I mean, and even if you do get those reactions, I mean, generally you just get treatment for them at the vaccination center. They'll give you a shot of adrenaline. Maybe for future you probably shouldn't get vaccinated. You're one of those people that can't be vaccinated. But like, guys, how could that keep you from getting vaccinated? I do not get it. We just have observed very, very few side effects from vaccination. It really is a miracle. Anyway, just something to consider. I have no idea where this anti-vax community has arisen out of. But it seems to really be picking up steam for some reason. I have no idea why. Anyway, that's all I'm going to say about vaccination. You guys get how it works, I guess, in general. And if not, let me know. We'll talk about it more. But I wanted to talk about which illnesses we generally vaccinate against. Because you guys have been vaccinated for stuff before, almost certainly. You've probably had your MMR. You almost certainly have had your MMR vaccine, measles, mumps, and rubella. And that's not all. We have a fairly aggressive vaccination schedule for babies and for young people for good reason. So you probably don't know what they are, though. You've had those vaccinations, but you probably don't know what we actually immunize against. So I want you to check at some point while you're working on your own here. What vaccinations do we give people and what is available? Okay. So you can just list that in there. It's a fairly big list, but you just throw in some vaccinations that you probably received. But what do we give people and what's out there? I'll give you a really cool example. You never have to have chickenpox, guys. You can get a chickenpox vaccine. When I was a kid, that didn't exist. I got chickenpox. I have scars still from having chickenpox when I was a kid. And now when I'm an elderly person, I am at risk of redeveloping chickenpox in a form called shingles, which is very painful for the elderly, which is unfortunate. But you never have to have that. You can get the chickenpox vaccine and you don't ever have to get chickenpox. Unless you've already had it and that's too late. But it's a possibility. And consider that. I mean, it's really a miracle. These illnesses are terrible. So, how do they decide which vaccinations are required? Another great question. Public health decides what they believe to be the essential set that is necessary for. So by the way, if you've had shingles trend, that means you've already had chickenpox. And unfortunately you can get it again, which is too bad. But, young people can get it too. Yeah. If you have a period of your life where you have a depressed immune system for whatever reason, even from stress, people can get shingles. It's just much more common in the elderly. Sorry, Michaela, I didn't mean to jump off your question. Essentially, we decide what are the most deadly illnesses for young people that are preventable. And if they are of enough of a public health concern, Health Canada decides this in Canada, but they decide which vaccines are essential for young people to have. For example, if we didn't vaccinate against measles, we didn't make that a mandatory vaccination, I mean, we would constantly be having outbreaks of measles. And people die from the measles, so there's that. But it can also be crippling for young people. It can cause sterility in men. And you don't have to have measles. There's no reason for that. So, to protect public health, we've essentially made it mandatory. You have to get those vaccinations if you want to attend public schools, if you want to be a member of the community. There are very little to no side effects from getting these vaccines. So there's really no reason not to get them. But anyway, we decide on what is an essential set. Chickenpox has not been made part of that essential set for young people, but it may in the future. Keep in mind that we have a publicly funded healthcare system in Canada, and so it is in our best interest as taxpayers to vaccinate people, because the medical treatments that we have to give to people when they get these illnesses costs money. And it's much cheaper to vaccinate people than it is to treat people that get these illnesses. So, and not to mention the effects of getting these illnesses can be quite terrible. HPV is a great example of one that we've added. Now, it's optional. I have no idea why this one is so controversial. I think it's because HPV can be an STI. It isn't always. You can get HPV not transmitted as an STI. But it is often an STI, and whenever the word sexual comes up in the context of government or public health, there's always like a weird thing that's placed on top of it. People don't want to, the controversy here is that people don't want to vaccinate against it because they're afraid they will encourage young people to have sex, which I think is ridiculous. But anyway, I think that is the concern to some. And so there is some controversy surrounding it. It's not, I don't believe it is mandatory, but I mean, there's no reason not to make it mandatory. It prevents cancer. And really interesting to think about why it causes cancer. The virus manipulates the DNA of the lining of the uterus, and which can potentially cause cancer. That's cancer is modifications to the DNA that cause cell replication to happen uncontrolled, which you probably would have talked about in grade 10. But so we can just prevent that. Almost all cases of that cancer are caused by HPV. So like, why you wouldn't get that vaccine? It's like, absolutely, I have no idea why you wouldn't get it. But maybe your parents didn't let you or whatever, for whatever reason. But anyway, let's just say that in a perfect world, we would vaccinate people against that always. There's no reason not to. And there are a number of vaccines that may get incorporated into the essential set. I think chickenpox may be heading in that direction. So yeah, so it's based on newly discovered risks. That's why you get the HPV vaccine. So you're right there. Why do you think there were many people against the vaccines in schools, but there's so many against COVID? Great question. Confusion, lack of scientific awareness, maybe the COVID vaccines are relatively recent, and we haven't studied the long-term effects of, for example, mRNA vaccines, although we have absolutely no reason to think that they have any kind of different or negative long-term side effects. We don't have the same long-term experience with this vaccine. It wasn't rushed. We did do the trials very quickly, but only because we threw a whole bunch of money at it and we were able to organize those trials so rapidly. The length of the trials and the integrity of the trials is as high, or in some cases much higher, than they would be for any other vaccine. Really, public health organizations have tried to be so thorough with the COVID vaccine because it's so important to have public confidence in it to get a high rate of vaccination. So, no, you are doing the analysis questions. This particular analysis question, page 59, number 15, you don't need to know. You don't need it to do, and it is not listed on Brightspace either. I always list the analysis questions on Brightspace, and I have not listed that one. That it's not a great question, and I removed it. Anyway, I'm not 100% sure why people are so against it, but I think it has to do with the relative newness of it and people's distrust in science in general and people's distrust in the government in general. But certainly a lot of work has gone into very thoroughly testing these vaccines. So I certainly don't think it's warranted, but some people may. That's right. You're exactly right, Michaela. They did take research from the SARS vaccines in the 2000s. And mRNA vaccines themselves are not new. There are other mRNA vaccines that have a long history. They're just not big, mass-distributed vaccines that have been given to hundreds of millions of people, which we're about to do with this vaccine. But that's not to say that they haven't had a long history and have been thoroughly tested. So again, I think people are confused. People don't know about the actual history. Which is leading them to make misinformed decisions regarding the vaccine, which is really unfortunate. And that's even true for people in my family. Man, I could talk about this with them all day. And it just, for some people, it's just a gut reaction. I don't get it. But anyway, I'll move on. Yes, I agree with you, Ryan. Yep. I mean, it was rushed in terms of they tried to get the process done as rapidly as possible. But they didn't change the thoroughness of the process at all. The number of people that were monitored and the length of time that they were monitored is the same as any vaccine approvals process. That was not rushed. That part was not rushed at all. It's the administrative part, getting started with everything, which normally takes years for a vaccine. All of the pre-administrative stuff and getting approval to start and all that normally takes years. And so that stuff was rushed. Let's get started stuff was rushed. The actual trials themselves and how long they're monitoring people and the analysis of the data is every bit as thorough as any vaccine ever. So in some cases more thorough. So people honestly have nothing to be concerned about regarding that. You can read the research papers; they're in public domain. Like you can see what the methodology was. It is very thorough. You can make a comparison to the flu vaccine to any other vaccine. You'll see that it is a very thorough process. It's nothing was skipped. So which is why it took so long. People are like, oh, why is this vaccine taking like a year? And it's like, guys, it's taking a year because they did all the things that you're supposed to do to make sure it's safe for people. So anyway, that's neither here nor there. I'll stop talking about this now. You guys have work to do. So don't worry about any of the protist stuff from today. We're gonna move that till tomorrow. Going back to what you're going to be working on now, there are some questions as we were going along that were in the course notes. They're little research questions here and there. I'm going to get you to do those. Normally I don't talk this long during this day, but there's been really, really good questions here, guys. I appreciate the discussion. There's a summary here of prokaryotes. I believe it's fairly short. Yeah, it's about 5 minutes, 6 minutes long. It goes over most of the stuff that we covered in the note. But with a little bit of extra and some examples. The TED Talk about quorum sensing. If you're absolutely pressed for time and you want to cut something, you can cut that TED Talk. There's nothing in the quiz about the TED Talk. It is very fascinating, though. So if you're interested in microbiome and you're wondering about how bacteria communicate with each other, it is absolutely fascinating. So I recommend it to you, but I would say skip it if you're pressed for time. I mentioned that there's question 4 in the course notes. That's in the course notes as we were going through it. And then these analysis questions go in your homework document. It's 3 and 14. There's just 2 questions. Okay, then we did the lesson on viruses. The first lesson here talks about the lytic cycle and the lysogenic cycle, which is how viruses reproduce. There's 2 ways that they reproduce, which we didn't cover in the lesson. So I highly recommend that you watch this video. This does have some important stuff that is on the quiz, specifically about the lytic cycle and the lysogenic cycle, which are how viruses reproduce. So that's the first one here. The second one is not on the quiz. So this is where do new viruses come from, but it is fascinating. If you're interested at all about how a new virus can appear, and I think generally a lot of people are interested in this now, how is it possible that a new virus can appear out of nowhere, like COVID-19? Great video that explains that process in detail. And it's not too long. I think it's about 5 minutes long. Okay, there are a couple questions in the course notes about viruses. I pointed them out as we went along. And there is a optional article on autism and vaccines. If you're curious where that controversy came from and why people are worried about autism and vaccines, and those studies that have been totally debunked are still cited constantly, to my great annoyance. If you're interested in that controversy at all, there's an article there about it. Okay, so that's what you guys are going to work on. If you sent me a question, I will definitely get to it. Anastasia, I'm just going to answer your question real quick here before we leave. Should we make vaccine recipes public knowledge? They are. So the actual ingredients that go into the vaccine, what is used as a preservative, what is used as attenuation agent, which means that it triggers helps trigger an immune response. Aluminum is usually used for that. That is public knowledge. So there's nothing proprietary about what is physically in a vaccine. It has to be public knowledge because there is a risk of allergens being in it. So you have to tell people what is in it. Like, for example, if you're using some kind of egg proteins, you have to tell people that, so if they're allergic to eggs, they don't get your vaccine. Now, they would never use egg protein in a vaccine like the COVID vaccine because it would make a huge percentage of people ineligible to get the vaccine. So what they try and do is use things that are essentially almost completely hypoallergenic, things that do not typically cause allergic reactions in humans. And that's to increase how many people can take the vaccine. But they are public domain. You can look at what the recipe is. Now, the exact process to make the vaccine is proprietary. How do you take a piece of RNA or a piece of DNA and then reverse engineer that shape and then build the antigen using that shape? That's proprietary knowledge, which I understand. These companies paid, in some cases, hundreds of millions of dollars to develop the vaccine. That process is their development. They own that, which fine. That's how business works. That's capitalism. That's the system we live in. So I don't think that you can necessarily ask a company to do that, to reveal that. But I will say that the general process for how to do that is public domain. How do you take a sequence of mRNA or RNA or DNA and then build an antigen from it? That's all public data. So they're doing that at universities all over. That's all public data. But the exact process that they use for a specific vaccine, for example, amounts of everything that they're using in their particular experiment, is generally proprietary. So, but it's reproduced all the time. Can another person or another group or another company make their own mRNA vaccine with what's publicly available? Absolutely. 100% yes. And people are doing that. So there's a very small element of it that's actually proprietary. I'm not sure if that answered your question, but it's not a secret. There's no secret stuff in vaccines. There's no microchips. Bill Gates is not involved. Yeah, you don't need to worry about there being anything hidden. You can look this up if you'd like. I will find a paper for you. I will show you what the components are in any particular COVID vaccine. I'm happy to show you. It's not proprietary or secret. You would like to see that? I'll find the information for you. Is that a yes? Okay, cool. I'll find that and I'll put it up on Brightspace, and I'll leave a note saying that I've added it to the end of the day. You just probably need to refresh day 3 once I throw it up there. I can do that for you, no problem. Okay. And then I'll leave it at that. So if you guys need anything from me, I'll be here. You can just post stuff in the chat or drop me an email. All right. Good luck. Sorry we're talking so much.